the planet we're used to today was most likely extremely different a billion years ago, was even more different 2 billion years ago, and most likely changed its appearance every once in a while. For example, we know that a few billion years ago, it's quite likely Earth was purple, or at least the oceans made it appear purple. This is usually referred to as the purple Earth hypothesis. You can learn more about this in the description below. But during this time, photosynthesis was very likely based on retinol, making the water appear this way. This is from Lake Hillier, one of the pink lakes in Western Australia. Then, about 1.8 billion years ago, Earth experienced a major glaciation period we sometimes refer to as the Snowball Earth. This was actually just one of several such events. During this time, a lot of biosphere on the planet seems to have reset, and once the ice melted and we found ourselves with a new Earth, this began a completely new era. And because of the presence of photosynthetic bacteria that relied on hydrogen sulfide instead of water, and actually produced sulfur instead of oxygen, the color on the planet and the color of the oceans most likely changed once again. Here, some scientists believe it might have even been black, or possessing some other unusual coloration, even making it appear slightly milky instead of blue. And strangely enough, today we believe that Earth remained this way for at least 1 billion years. But we actually know very little about this period. For one simple reason: not a lot seems to have happened for that 1 billion years. Not a lot of geological activity, no major volcanic eruptions, no powerful impacts or asteroid collisions, and even stranger, the plate tectonics or continents did not change much either. It's as if planet Earth decided to just take a break for 1 billion years. And so when looking at fossils, this is actually one period that we basically know practically nothing about. Today it's known as the boring billion. The strange period on planet Earth where not much seems to have happened, and even the life itself did not seem to change much either. Or at least that's what we used to believe up until, I guess, the last few years. And, well, hello wonderful person, this is Anton, and today once again we're going to be discussing incredible and completely unexpected discoveries about that boring billion. Once again, there's a completely unexpected discovery, and here I think pictures will probably do it justice a little bit more, that in essence show us a ridiculous diversity of life that seems to have existed back then, but that we practically know nothing about. And all of this once again discovered completely by accident. With some of these life forms resembling something out of a science fiction story, not so much anything from modern life. But here there is quite a lot of evidence that this does seem to be somewhat complex life, possibly even similar to modern mushrooms. Or I guess more specifically, fungi. And so let's discuss these two incredible studies and what was discovered so far, and of course let's talk about what this might mean for our understanding of evolution of life on our planet. Okay, that last question is pretty easy to answer. We once again just realized that we know very little about how life evolved and how complex it already seemed to be approximately 1.8 to 1.5 billion years ago. And so even when it comes to trying to understand when some of the more complex cells evolved, there's still a lot of confusion and just not enough evidence to determine anything. But if you've been watching the videos for the last few months, you know that this is not the first video about the so-called boring billion. As a matter of fact, we haven't talked about this topic for many years, and suddenly there are several studies discovering something absolutely incredible about this period. You can find the links in the description, but in a nutshell, there were two major discoveries. First, it was confirmed that the rotation of the planet was approximately 19 hours per day, but intriguingly, it hasn't changed for that whole billion years. This was completely unexpected, but the evidence seems to be there. But second of all, there was evidence for an unusual life or unusual biome that seemed to exist prior to anything that's known to modern science. It's actually completely unknown what this might have looked like, but the signs of this life seem to be there based on molecular analysis. And it seemed to be everywhere on the planet, but was definitely different from any modern life and from any modern cells. Nevertheless, it seemed to be dominating the planet and was probably a large part of the biomass. And it's that last discovery that kind of takes us closer to, well, the big mystery. How exactly did the modern cells form? Eukaryotes are way more complex than bacteria or prokaryotes. But exactly how this evolved compared to this is not a very easy question to answer. Okay, we don't even know how bacteria evolved, but how bacteria then somehow transitioned into eukaryotes is an even bigger mystery. And so, discovering these unusual ancient organisms existing during the Boring Billion could provide some of the answers. Maybe this was the transitional stage. But that particular study had an obvious problem. The only evidence was molecular, and no actual fossils were ever found. And that's actually the biggest problem with studying the Boring Billion. Because the Earth was so quiet and there was no geological activity, there was also not a lot of fossils. And so we don't actually have a lot of fossils from that period showing us anything. There are fossils of bacteria and there are certain fossils resembling some kind of an algae, but they're extremely rare and generally do not show much. And so up until I guess this point, it was almost impossible to imagine what any of this life would look like. And then we have this new study. And once again, this was discovered completely by accident. The scientists studying this weren't even biologists. From what the scientists described, they were actually studying rocks, specifically beryl and topaz. And here they gathered this from one of the Ukrainian mines that's known to contain quite a lot of different gemstones. This mine is known as the Volyn mine, and this whole discovery is now known as the Volyn biota. But because they were actually studying the structure of these rocks using advanced electron microscopy, they then realized that completely by accident, a lot of these samples seem to also contain strange three-dimensional shapes. Shapes resembling something they've never seen before, and actually resembling what we might describe as some kind of primitive life. With I think this one probably being the coolest, and also being the one that's kind of difficult to explain. With of course the biggest question now being, so is this what that life looked like? Is this the life that was just described in that other study from just a few months ago? And is this the representation of the transitional stage between prokaryotes and eukaryotes? So right now the answer is, I don't know. But I'm sure many of us want to believe this. These are essentially the oldest three-dimensional microfossils ever discovered. And this is like 1.5 to 1.8 billion years old. This was actually present inside a relatively thin layer of aluminium silicates present inside various minerals. But even stranger, a lot of these appear to be multicellular. And some of them do appear to be quite complex. Here it was described as the fungi-like organism, but some of them also seem to contain some kind of a shell around the cells. But more importantly, a lot of these structures have never been seen before in any life. Definitely different from anything we observe today, or from anything in the last billion years. And in terms of size, some of these are pretty small, just over 10 micrometers in size, but some of these were large. Quite a few of these were at least several millimeters in size. The ones with filaments and with multiple structures seem to be the biggest. But exactly what any of this is anyone's guess. These are literally the first ever skeletons preserved in ancient samples that we can physically see and observe in 3 dimensions. Pretty much all of the other samples are usually from much younger animals, such as the ones from Cambrian explosion. And one of the main reasons we don't really have any samples from before is for one simple reason: skeleton. Anything that does not have skeleton is very difficult to preserve in fossils. Which is why all of the previous discoveries were just based on chemistry or based on interaction with the environment, not so much actual physical shapes. There were some, but they were extremely rare. But now we have physical representation of something that existed on a planet almost 2 billion years ago, and all of this preserved with relatively high detail. But I guess the question is, so what do we actually know here? So do we know anything about this life? Well, the only thing the researchers seem to be pretty certain about is where exactly this life might have existed, or basically what sort of life this was. And it was most likely terrestrial life, but not surviving on the surface, instead living inside some sort of a geyser-like structure, possibly hundreds of meters below the ground. And this geyser system potentially contained all of the necessary biological components required for the growth and proliferation of these organisms. So here they were basically surviving inside some kind of a cavity, possibly as deep as 2 or 3 kilometers. But because some of this water was extremely rich in silicon fluorite fluids, once the liquid started escaping to the surface, it almost instantly fossilized a lot of this life, while also creating all of the minerals the scientists recovered. With the main implication being that a lot of this very likely existed in extremely deep underground habitats, with many of these microorganisms very likely surviving and thriving right between the rocks. With one implication being that maybe this was not the most common organism that existed back then, and possibly just represented some kind of a niche, but because we actually find life in similar conditions on Earth today, comparing both in some of the future studies might provide some answers. At the moment, because this is just a brand new discovery, other than the actual pictures, there's just not much else. But the pictures themselves are definitely super cool. Just the fact that this survived for almost 2 billion years and is still visible today is already quite impressive. And because now we know where to look for more similar life, there might be more incredible discoveries from this unusual period in the next few months. Hello wonderful person, this is Anton, and today we're going to be discussing some of the new advances in regards to the idea of artificial life. Life created completely from scratch. Very often combining our understanding of genetics with essentially our understanding how various cells form and how they eventually end up as various types of primitive life. And in this case, we're going to be focusing on what you see right here on this screen, the creation of artificial life that can now move around by essentially acquiring new genes. But in order to understand this achievement, and in order to understand what the scientists actually created here, we first have to take a few steps back from this discovery and briefly discuss some of the other discoveries from a few other videos you can find in the description. Now, this idea of artificial life, or trying to create some kind of an artificial cell, is something that actually started to kind of explode in the last 10 years or so. And the biggest project that kind of deals with this, that we discussed in one of the videos in the description, is a project known as JCVI. The project that for the past decade or so focused on creating the first ever artificial cell capable of performing basic functions of life. And this project only started approximately 12 years ago. As a matter of fact, the first version of this particular cell was originally built in 2010, with the major goal of this synthetic biology being the capacity to predictably design and build various types of DNA that can then actually end up producing a cell. And though the initial steps here are to basically try to create an actual artificial cell that's able to function just like any other cell that we have around us, eventually they want to actually find a way to even create new biological functions as well. And in order to make this work, the scientists behind the original study did something relatively clever. First, they tried to find the bacterium with the smallest known genome. In this case, they decided to focus on Mycoplasma mycoides, the parasitic cell that contains one of the smallest genomes from the known bacteria. But once the genes from this cell were identified and mapped, the scientists then started to essentially knock out some of the other genes in order to find out which genes need to be left behind in order for this organism to actually function. Or in other words, they basically wanted to remove any other additional genes present inside the cell, leaving an absolute bare minimum that can then result in a similar cell with potentially similar functions. And eventually they've discovered just over 450 genes that were absolutely crucial for the cell to function. Everything else though was more or less redundant, which resulted in the production of the first ever synthetic organism. But more and more advances have been made in the last few years, and the scientists started to push the envelope. First, they actually tried to find a way to have this organism replicate, which is what I've discussed in that previous video you can find in the description, while also discovering additional genes for a lot of other functions as well. In other words, they've created a kind of a bacterial Frankenstein's monster, an entirely artificial organism created from individual genes collected from several different cells, capable of performing certain functions very similar to a typical cell, but not all functions. As a matter of fact, this is still a work in progress. And quite a lot of new advances have been made in just the last few years. As you can see, we're now on version 3.0, and this organism, sometimes also referred to as Synthia, to some extent has been evolving really quickly. Although in this case this is entirely artificial evolution. But in the last 3 years, a lot of this has been actually advancing pretty quickly because of computational modeling and all of the publications that made the entire genetic catalog from this particular cell available to essentially anyone, identifying all of the essential, non-essential and quasi-essential genes, as well as giving comparisons to other organisms such as, for example, the famous E.Coli. And so because of these advances in modeling and advances in genetics, it's now become a lot easier to modify the cell even further. And though last year we've discussed the advances in reproduction, this new advance coming from Japan involves motility, or basically moving around. And the thing about motility or moving around is that It's actually a really complex process, and it looks like a lot of different organisms evolved entirely different ways to move around using entirely different sets of genes. And moreover, a lot of these motility tools, for example cilia that a lot of bacteria possess for motion, were eventually evolved into entirely different, very important functions in much more complex animals like us. For example, this is an image from our lungs. And in this case, these same type of structures are essentially responsible for protecting our lungs and for maintaining our airways in order to allow us to breathe much easier. And so obviously understanding how this was created and how it evolved in various animals is also important for a lot of other medical reasons. But in this particular study, in this particular case, it represents the first step in understanding how motility might have evolved in simple life and what genes are responsible for creating simple locomotion. But interestingly enough, even though so many different cells learn to move around using various methods, even today it's still not entirely understood how some of these functions form and what genes are responsible for the formation of certain structures. But in this case, the synthetic cell, Cynthia, was essentially a perfect way to study this. By default, Cynthia does not move. It's a non-motile cell, and it doesn't have any genes or ability to move around. On the other hand, one of the simplest bacteria that can move around is known as spiroplasma. It's believed that the motion here is achieved by basically shifting around the structure of the cell itself. Or in other words, by switching around the shape of the helix, and by twisting and turning the outer cell wall, this bacterium can then perform a basic function of moving forward or moving backward. And so the idea here was to take some of the genes possibly responsible for this function and to then insert them into the artificial cell Cynthia, just to see what happens. With the obvious goal being a potential swimming organism that can basically switch around its cytoskeleton in order to propel forward or backward. Now prior to this has never been done before, and the previous motility was extremely basic and basically was not organized at all, and so this was definitely the first attempt to create a moving bacterium. But it didn't work right away. As you can see, even though spiroplasma moves around just fine, and Cynthia doesn't move around at all, the first versions of modified Cynthia were not moving either. But eventually they've discovered that you really only needed to have 2 genes, or basically only 2 proteins that would be required for motility of this cell. And so basically by removing all of the potentially redundant genes, they were able to create the first ever minimal motile cell, or essentially artificial cell that moves around by itself, with the name of the protein responsible being MREB. But intriguingly enough, this protein does exist in a lot of other bacteria or other cells, but in many other cells it doesn't seem to create anything, or at least does not create motility at all. And so this of course implies something really important. There are definitely multiple different evolutionary pathways that can create a similar function, with the same gene not necessarily creating the same functions. In this case it definitely created the necessary motion, but if you were to place the same gene in a more complex cell, it's unlikely to have the same effects. Which is why the team behind the study is already trying to find other genes that can actually create similar effects. And to be honest, these discoveries are actually important for so many different reasons. Not only the reasons of understanding how evolution works, but just the overall idea of creating artificial life with functions that you wanted to have. Or basically think of it as a kind of a sandbox Lego, where you have all of these Lego pieces, which are your genes, that you can insert into an artificial cell in order to then create some kind of an artificial bacterium that does exactly what you want it to do. So basically kind of a genetic Lego. But obviously these are all still really early steps, which of course means that we'll be hearing more about Cynthia and a lot of the discoveries of artificial cells for many years to come. This is honestly one of the most fascinating biological studies that could one day lead to some major breakthroughs in genetics and of course in evolutionary biology. A few months ago, we've talked about discoveries in regards to synthetic life, living organisms created entirely by combining DNA and the cell structure from various species, in a sense representing a kind of a Frankenstein monster cell. And in the last video that you can find in the description, Japanese scientists were actually able to add genes to make this artificial cell now move around. But in this video, we're going to be discussing this new study that as always you can find in the description below, that in a nutshell discovers something else really unusual about these synthetic cells. But I guess first, well, hello wonderful person, this is Anton. Let's discuss Cynthia once again. The unusual bacterial organism developed by reducing DNA of a known bacteria eventually turning it into what the scientists now refer to as synthetic organism. Something that, even though it might sound controversial, actually is really not, mostly because of the reasons for why this was done and what the scientists are trying to learn from all of this. And the idea here is really simple. Take a bacteria with the smallest possible genome, or really any organism. It doesn't actually have to be a bacteria, but bacteria generally have the smallest DNA of all organisms, and then essentially remove anything that doesn't seem to be useful, reducing DNA to the absolute minimum in order to see if it can actually still create an organism that you started with, or at least something similar. And so here the main point was to reduce a living organism to the absolute essential genes, with only one main reason: trying to understand exactly what's required genetically in order to build an organism completely from scratch. This actually even has a name now. It's known as Mycoplasma laboratorium, because it's basically a bacterium produced entirely in a lab. But the early research that started a couple of decades ago began with some of the bacteria with some of the smallest genome. At first it was the bacteria known as Mycoplasma genitalium, something that's actually sexually transmitted and something that many of us do have in our bodies, depending on how naughty we are. But because it has a relatively small genome, it was a pretty solid first target. But eventually several teams realized that this is a much better target. This particular bacterium is even simpler, and in essence is a parasite living inside the intestines of various goats or a lot of other similar animals. And intriguingly, parasites in general, because of their lifestyle, tend to lose a lot of genes that are no longer required. And that's because once they find a lifestyle that seems to provide them with everything, they no longer need a lot of other functions and thus eventually lose a lot of genes. And theoretically, bacterial parasites would have the simplest genome out there. And that's exactly what the scientists discovered here and why they decided to use this particular bacterium. Bacterium whose genome is actually really small. It contains approximately 901 identified genes. But the thing is, the majority of these genes are still unknown to us. We don't really know what they do. And so, through sheer trial and error, by removing each of the genes individually, at some point, various scientific teams were able to remove 41% of all of the previous genes, with just 473 genes identified to be essential for this bacterium to function and to survive. So basically, half of the genes here were surprisingly not needed at all. It's unclear if they were totally not needed, but the bacterium was doing just fine without any of these genes. But interestingly, even here, there are quite a lot of unknown genes, or genes whose function is not understood. Also, just to give you a comparison here, today we believe human genome contains approximately 100,000 genes, but only 6,000 so far have been definitively identified. So here, this is definitely a lot less. And so 150 of these have function that is not really clear. Nevertheless, because of this organism is now officially known as first truly synthetic life out there. But now we know that even smaller genome is technically possible by using something with an even smaller genome. For example, here we have something with only 169 genes. And intriguingly, what makes this synthetic and artificial is the fact that everything here was constructed artificially by adding various genes and the cytoplasm from other cells, and then putting everything together kind of like Legos. In other words, they knew exactly what genes to take, they knew exactly what parts to take, they put it all together, and it seems to have worked. But all of this was done a while ago, several years ago as a matter of fact, with an older video explaining all of this. And as you might learn from that recent video from just a few months ago, now the scientists have also started adding a lot of other genes, including genes for motility, allowing these cells to move. So basically now we have a moving Frankenstein's monster, or I guess bacterial Frankenstein's monster. But you can actually learn about all of these new advances in the link in the description, with a website essentially dedicated to all of the new advances and new experiments, and of course new studies about this strange synthetic organism. But now we have a new discovery that's kind of difficult to explain and is definitely unexpected. It's coming from this paper, Evolution of a Minimal Cell. In essence, testing something really simple and something that we thought we understood pretty well. Evolution. Specifically evolution in regards to mutation of different genes. And this is of course based on a lot of different experiments from the past, where a lot of this has already been done with a lot of other bacteria and a lot of other species. Here the main concept is gene selection. or essentially the genetic variation responsible for certain types of selection in order for the stronger organism to eventually survive. With I guess one famous example being the Darwin's finches. Slightly different genes, responsible for the slightly different beak, resulted in different types of finches dominating certain types of environments on the Galapagos Islands. So for example, if a bird had a much bigger beak that would be much better for opening things like seeds, that type of gene would become prevalent in this particular environment. And this also relates to another intriguing concept known as the population bottleneck. In essence, some kind of an event, for example some kind of a catastrophe, that dramatically reduces genetic variation, even potentially leading to extinction events, suggesting that an organism with more genes and obviously more variability is more likely to survive a certain event. And in some sense, by creating this synthetic organism, you could almost see this as a kind of a population bottleneck. We have this new organism that has approximately 50% less genes compared to a similar bacteria from a similar environment that was not genetically modified. And so at least on paper, it would look like the synthetic organism would not really be very adaptable and would not be able to mutate very well. As a matter of fact, it might even seem like it might be prone to extinction much easier, because some of those genes that were previously removed might have had some hidden function, such as providing some survival benefits. And so at least on paper, if we were to put both of these organisms, the natural one and the modified, in the same environment, the assumption here would be that the natural organism would most likely outcompete the synthetic organism in most situations, especially when it comes to more difficult conditions or conditions more difficult for survival, since the synthetic organism just doesn't really have a lot of genes that could provide additional benefits. But that's on paper, that's what was assumed. And that's what the scientists behind this paper wanted to test. They wanted to find out if this is actually what's going to happen. Turns out, it was the opposite. And that's of course the surprising part. Here they introduced the Syn3b, the third generation of this organism, and the original cells that were not modified, to various challenging conditions for approximately 300 days. This would be equivalent to about 2,000 bacterial generations, or in human terms that would be like 40,000 years. They then essentially allowed these organisms to do their own thing, and check the results afterwards. And to their surprise, compared to the original, the new strain was evolving approximately 40% faster. And on top of this, it didn't just evolve, it seemed to have regained all of the fitness that was lost previously when many of the genes were removed. And this is where I should be playing that Jurassic Park theme and quoting Jeff Goldblum when he says, "Life finds a way." Because that's what seems to have happened here as well. For reasons still not understood, when placed in exactly the same conditions, after 300 days, the synthetic cells outcompeted everything else, becoming the dominant strain and taking over the entire environment. And exactly why this is happening is anyone's guess. Theoretically, the complexity of other cells should give them a bit of an advantage, but it clearly doesn't. Instead, giving the advantage to the synthetic cells that are able to do everything much quicker and much more efficiently. On top of this, you can actually see the main difference between these cells, which once again really surprised everyone: the size. The original cells are actually able to grow in size because they have all of the genes. The artificial cells are missing some of the genes responsible for the formation of membrane, and because of this they cannot expand their volume and essentially maintain their size. And so even though it was preferential to grow bigger in size, these synthetic cells somehow found a way around it and managed to create all of the function needed without growing bigger physically. And so yeah, all of this so far, all of these discoveries are to some extent, at least for biologists, kind of mind-blowing. Mind-blowing meaning. Especially because at the moment it's not clear what genes were evolved, why they evolved so quickly, and what exactly is it about the natural selection that we still don't understand, especially coming from this very unusual example. Because it essentially suggests that the simplest organism was able to somehow optimize much faster and eventually take over the entire environment. Or in biological terms, it became a lot more genetically fit even though it technically shouldn't have. In essence, regaining all of the ability that were lost when the genes were artificially removed. How it was able to do so is anyone's guess. Okay, I might have one guess, but it's a very long shot here. From a lot of previous research, we know bacteria are generally also very good at stealing others' genes. They're basically able to share and recover genes that are often found in the environment. And so because all of the bacteria were in the same environment, there's maybe a slight chance for some kind of a genetic mixture, with the synthetic bacteria eventually recovering some of the function by basically consuming the genes from other bacteria. It's a big guess, there's no proof yet, but at least for now that's maybe one possible explanation. And so here it would be very important to test if these new bacteria, the synthetic bacteria, still had the same small genome, or if they actually changed something, adapting in the process, and basically consuming things from the environment. We'll probably learn more about this in some of the future videos. In the last few years, we've had quite a lot of different advances in biology and quite a lot of really interesting experiments showing us things like synthetic life, synthetic cells, and even synthetic cells that learn how to reproduce. With many of these experiments previously mentioned on the channel, and that can also be found in some of the links in the description. With even things like potentially printing an actual body part, 3D printing that is, becoming a reality. For example, earlier in 2021, scientists demonstrated an ability to print human bone of a specific shape using real biological cells placed inside a gelatinous bath that could then be used in surgery almost right after it was printed. Then, a few months ago, also in 2021, using a completely new laser technique, scientists managed to 3D print functioning mouse brain cells with a technique known as laser-induced site transfer, and this fabricated tissue-like constructs by delivering all of the material in layer-by-layer process similar to commercial 3D printing. In this case, they actually used a type of a neuron cell to create a Bioink, and even though it was not a perfect reproduction of an actual mouse brain, it was still a pretty good first step. Although, the only known example of a 3D printed body organ that still functions in a human today is a bladder that was grown from an actual biological tissue and was then implanted into a child and still functions in him even today. You can read a little bit more about this from the article in the description. But 3D printing an organ is still extremely difficult to achieve, mostly because it requires extremely precise printing of actual blood vessels, and that's something that most scientists still don't really know how to achieve. And so the 3D printing of human organs, or really any organs, is still in its infancy. It still has ways to go before it can become a practical way to solve a lot of problems. But printing much smaller tissues and much smaller samples of organs is a reality, and it's already sort of become a really important reality because, well, first of all, it has dramatically decreased the need for animal testing. For example, using animals to test a certain product, to test cosmetics, to test different types of medicine, has been slowly replaced by these 3D printed organs in various industries. Here's actually an example of so-called lung on a chip that serves as a model of human lungs in order to test the drug safety of drugs being developed for treatment of human lungs. And it actually works really well and obviously avoids the controversial issue of animal testing. But now we have something a little bit different, something to some extent even more advanced. The scientists whose study you can find in the description below came up with what they refer to as the Bioink. The ink that then allows them to literally 3D print anything. And the ink that for the most part kinda acts like the typical filament you would find in a commercial 3D printer. But this ink is made entirely out of living things and can in theory be used for a lot of different things, with scientists already demonstrating that this ink can first of all create structures that can then produce medicine, also creating ink structures that can then absorb toxic elements, and even suggesting that you could technically build some kind of a structure out of this that can then be self-repairing. In other words, as the title implies here, they've created a programmable ink for 3D printing made out of bacterial cells that can be easily genetically modified to do a lot of things, including things that they obviously haven't even thought of yet. And here the premise is really simple. We know that a typical living cell can produce a lot of different things depending on what the genes tell it to produce. And so it becomes possible to engineer a cell that produces a certain component that then produces something else. Okay, let me explain this by using the example from the paper. So, for example, we can take an E.Coli cell, the most widely used cell in genetic engineering, and program it to produce a certain type of a protein. That protein can then assemble itself and create larger structures, such as, for example, these filaments you see right here. These filaments can then act as 3D printable ink that can then be used to build something even bigger.
But inside of these structures, inside of this ink or inside of these filaments, you can also program it to do something else. For example, absorbing something like a toxic element, or producing something like a typical medicine, or, well, really anything you can think of. And that's pretty much what they have achieved in this paper. But the question here is, well, what kind of a filament would even produce this, or what kind of a protein did they introduce into E. coli? And this is actually where the brilliance of the paper comes in. Here, the scientists use something known as fibrin. It's the protein located in our blood that you can see in this simulation right here that acts along with the platelets to form what's known as the hemostatic plug. Basically, a blood clot that you see right here. And fibrin usually does this by connecting to a lot of other things and by turning into a kind of a mesh. Basically, it creates filament naturally. And so by using this protein and introducing it into the E. coli, along with some other genes in order to actually have function, the genetically modified bacteria then created the parts needed to create all of these fibers, with the actual fiber assembly happening completely automatically. And then through the process of filtration, this would essentially create the microbial ink that could then be used for 3D printing of a typical structure, with some of the samples visible right here. But by then also introducing other genetic modifications to the bacteria, these structures also acquired actual function. So as I mentioned, one of them was able to produce certain types of drugs, other one was able to extract toxins from an environment, and here the scientists also suggest that it's possible to modify them to be self-repairable as well, or possibly even have an ability to reproduce and to create larger structures, which the scientists have already suggested could be used in, for example, space exploration to create some kind of a self-sustainable habitat on, for example, the Moon, the Mars, and so on. So basically imagine the habitat that can actually produce a lot of things that you need for survival, but by basically just doing it itself. And also have an ability to self-repair if something gets damaged. Now obviously, as you can see from the sample images, we're far from being able to do any of this just yet, but the samples they've created have already been able to have functional abilities. And so this particular invention does actually have a very strong potential to have some really interesting applications in the future. And because an ability to create various types of medical devices for various applications has always been a bit of a challenge, an ability to create these living materials has a potential to transform the medical industry in its entirety. Especially because producing very certain 3D shapes or 3D structures has always been a challenge. Maybe not a challenge in a commercial 3D printer, but definitely a challenge when it comes to medical 3D printing or just medical devices in general. And so this type of a 3D printing of biological structures currently presents a really exciting opportunity for the future of the medical field. Although personally what I would love to see is the creation of an actual living structure similar to what you see right here, but on a much grander scale. Something that could be maybe used for maybe habitat, maybe something else. So literally imagine an actual habitat that produces things on the inside that you can then maybe consume or maybe breathe, or maybe it also uses your waste for its own survival. Something that, even though might sound like science fiction, is basically implied in this study. By modifying certain genes inside the E. coli, it does become possible to maybe create something that actually interacts with us as well. But I guess baby steps. First, let's see where the scientists take it in the next few years, and what they can actually create from these techniques by using the fibers that they've already been able to achieve in the study.
Hello wonderful person, this is Anton, and today we're going to be discussing a new study that just came out a few days ago that proposes another unusual cycle on planet Earth that we've never known existed, but seems to affect the amount of biodiversity on the planet. And it does so every 36 million years. And so in this video, let's actually discuss exactly what the scientists discovered so far, what this potentially means for our understanding of planet Earth, but also talk about what exactly drives this cycle and what effects it has on the planet. But first, let's start with the basic concepts. The concepts of biodiversity and the idea of extinction. Something we always hear about in the news, but something that's not necessarily well explained. For example, here's an incredible map of terrestrial biodiversity on the planet. What this essentially shows you is the amount of various vertebrate species, or the amount of various complex animals, present in various terrestrial locations on the planet. As you can see, some of the highest biodiversity, which is in red and in orange, is essentially in parts of Africa, some parts of Southeast Asia, and of course, Brazil. Most of it is in Brazil. But in this case, it's not really telling us about the amount of animals, it just tells us about the biodiversity, the types of animals in that particular location. But generally, locations that are more hospitable to life will always have more biodiversity. So for example, in a desert, you're not going to find as much biodiversity as you would in a forest or in some kind of a jungle. But this doesn't actually tell us anything about the total biomass or the total number of animals, or even how successful animals are in these locations. As a matter of fact, some of the least hospitable locations on the planet often have way more biomass. For example, this Japanese island known as Nishinoshima is practically barren on the surface, but it recently had a volcanic eruption that created a huge bloom of various algae visible from outer space, overall increasing the biomass by a relatively large amount. Something very similar happened after the eruption of Tonga, with the total biomass, as you can see right here, being in hundreds of kilometers in terms of size. And so biomass by itself is not always correlated with biodiversity. And that of course implies that even during, for example, extinction events, which by definition is a sudden decrease in biodiversity on the planet, doesn't actually mean that it's going to change the biomass in terms of size and mass. All of these new conditions on the planet might instead quite often result in the opposite. They actually result in a huge bloom of a lot of bacteria or algae, or possibly some other animals that were previously just a tiny niche, invisible otherwise. But when it comes to extinction events, especially the so-called Big Five extinction events, we normally define them as the sudden decrease in biodiversity on the planet, with the most famous one being the one that killed the dinosaurs 66 million years ago, but the more recent one only happening approximately 34 million years ago and referred to as Eocene-Oligocene extinction event. The event that also involved a dramatic decrease in a lot of different biodiversity, especially in the marine life, but the event that doesn't have a very good explanation even today. And although two potential craters have been discovered that could have actually caused this, one in Russia and one in the US, they don't really provide enough evidence for any of this, especially because, generally, just a single collision will often not result in a major extinction. And so even though a lot of people assume that an extinction might be caused by some kind of an asteroid event or a large collision somewhere on the planet, in reality the evidence suggests otherwise. Especially because these two craters were most likely formed millions of years apart, and at least a million years before the extinction event as well. But in the last decade or so, the scientists developed even better time maps showing us biodiversity across hundreds of millions of years. Here's one of the better ones that basically shows us that biodiversity does actually change quite a lot pretty much every few million years. But more importantly, showing us that it almost looks like there's some kind of a cycle here. And because a lot of these events never actually had any explanation or an explanation that would be definitive, it basically presents paleontology with another mystery. What exactly is causing sudden changes in the number of species on the planet, especially if it doesn't involve something very obvious? Here we're talking about some kind of a major volcanic eruption or some kind of an asteroid impact. And that's sort of what this new study is about. They basically worked out a pattern that seems to suggest there is actually a cycle, and it's approximately 36 million years, with the cycle in this case driven entirely by the geology of the planet, specifically by the idea of plate tectonics and the increase or decrease in the ocean levels. And all of this was based on two things. First, a much closer look at the fossil record, focusing on biodiversity in the last 250 million years, but then also combining this with a simulation of geological motion or plate tectonics using the script known as G-plates that you can find in a description. And in the end, the simulation established that roughly every 36 million years, the plate motion on the planet results in a dramatic change in the sea levels, resulting from either increase or decrease of the amount of seafloor spreading that happens on the planet, which then surprisingly results in a kind of a cycle that changes the total depth of the oceans and the amount of water that's transferred into the Earth's crust, which can then produce even more volcanism. And so the motion of the plates and the spreading of the seafloor changes the ocean levels just enough to then cause dramatic changes on the surface of the planet, either increasing flooding or increasing the amount of deserts and the amount of dry land, with most biodiversity occurring when there is actually a lot of different shallow seas across the entire planet. But when the ocean depth changes and the shallow seas either dry out or become deep ocean, that's when we suddenly observe a decrease in biodiversity, which can also be seen as an extinction event if it happens in a lot of regions on the planet. For example, the spread of the seafloor across various oceanic plates creates larger and larger oceans, and thus decreases the overall water levels over time. Likewise, subduction zones reduce the total ocean floor as some of these plates are then swallowed underneath the continental plates, eventually recycling all of this in the process. And so both types of plate tectonics can actually vary the overall ocean levels quite dramatically, with the discovery here suggesting that it's also cyclical. And more importantly, seems to correlate directly with the biodiversity cycles of 36 million years. And though it's possible that some of the extinction events could be associated with this as well, at the moment none of this is definitive, and it just shows us that the actual cycles on the planet are just way more complex than we ever thought. More importantly, providing evidence that the tectonic plates have their own cycle as well, and they seem to influence the ocean just enough to then influence life. Now this is of course just one of many cycles. As a matter of fact, the team behind the study might have found a second cycle that's about 62 million years, but none of this is definitive just yet. As always, extraordinary claims require extraordinary evidence. Although I guess one important caveat here is the fact that this is mostly, at least for now, based on the fossils from marine animals, not from terrestrial animals. As a matter of fact, for all we know, maybe it has the opposite effect on the biodiversity on the surface. But because we don't have enough fossils to study any of this, it will be difficult to confirm this, at least for now. But assuming that the scientists behind the study are correct and discovered a new cycle, I guess the more intriguing question here would be, does this actually mean that EOG events, or the Eocene Oligocene extinction, might have been caused by this as well? And if this was a result of this cycle, with the event happening 34 million years ago, and affecting the biodiversity of a lot of marine animals on the planet, well, does that mean that we're in for another one in a couple of million years? Now, obviously, this is not something we can prove just yet, but assuming that the scientists are correct here, in about 2 million years, the planet might actually start changing again, reducing the overall levels of water, shifting the overall biodiversity once again. But I guess exactly how, or what exactly would happen to the planet, is not something we're going to know just yet. Nevertheless, a pretty intriguing discovery and a pretty intriguing study, and something we'll discuss more in some of the future videos. Hello wonderful person, this is Anton, and today we're going to be answering a relatively interesting question of biology. Does anything actually eat viruses as a predominant source of nutrition? In other words, is there anything out there that actually survives on eating viruses? And though it might not sound like a very important question, it is quite important for a lot of different reasons, and actually does provide solutions to potential mysteries. So here's one mystery. If nothing out there ate viruses, or if nothing else was destroying them to some extent, at some point all of the viruses in the ocean would actually destroy every single cell, leaving practically nothing behind. For example, we know that bacteriophages that sort of look like this are actually extremely efficient at destroying a lot of bacteria. They usually invade the cell and use its own machinery to then produce even more bacteriophages, which then reproduce, creating even more copies, with each capable of doing the same to other bacterial cells as well. And so with time, technically, these bacteriophages can actually destroy all of the bacteria in the oceans, assuming that nothing is there to stop them. Yet we know that this is not what happens. There are quite a lot of bacteria out there, and something is definitely responsible for destroying viruses, or for at least causing viruses to destroy less bacteria. That's of course one of the mysteries. Other, more simpler mysteries are just in regards to viruses as a source of nutrition. For example, going back to the bacteriophage here, technically this is a really efficient source of a lot of stuff. There are a lot of amino acids, nucleic acids, lipids, nitrogen, phosphorus, with all of this extremely efficiently assembled and basically ready to eat right away. And based on what life does usually and how it's able to adapt to anything and consume anything that's extremely efficient in nutrition, it's only logical to assume that there's gotta be something out there that eats viruses as well. Yet up until this point, these organisms have never been identified. There are lots of organisms eating bacteria, there are also bacteria eating other bacteria, and all sorts of interaction that's been observed on microscopic level, but viruses for some reason more or less remain sort of untouched. So does this make them the top of the actual food chain? Or is there something out there that does use viruses as a primary food source? Well, the answer to that question is yes, there is something that was recently discovered. But actually even before that, there is a video from a couple of years ago that actually explored another organism that was actually discovered to use viruses at least to some extent. It wasn't really eating them as a primary source, but it didn't mind occasionally eating viruses as well. And that was an important first step because, using a somewhat similar technique, the scientists have now officially identified the first ever known bacterium
that basically chooses to eat viruses and gets most of the nutrition from them as well. And this concept now has a name. It's known as the virovore. Sort of like an omnivore, carnivore, in this case, something that eats viruses, a virovore organism. But based on this recent study, only one such organism has so far been identified, and it only seems to eat one type of a virus, or at least one known for now. And this tiny organism is known as Halteria, something that was originally found back in the 17th century. And Halteria is what's known as a protist, essentially a complex cell that's not an animal, not a fungi, not a plant, but does possess a lot of complexity on the inside, including organelles and a nucleus. And this organism also possesses what's known as cilia, tiny hair-like protrusions that it uses to move around and to potentially also grab various food particles. And normally these exist pretty much in every major body of water. In this case, the actual samples were collected from some sort of a pond. Here's a slightly more detailed picture of what Halteria might look like if seen in a microscope. And so by taking a sample from a pond, they then added a pretty common virus known as the chlorovirus, that sort of resembles something like this, into the sample itself. And generally chloroviruses tend to infect certain types of plankton or certain types of single-cell organisms, eventually leading to their destruction and obviously a dramatic increase in viruses. And so here it was expected that this virus will eventually proliferate and destroy pretty much everything on the inside. But the scientists had this curious question: is anything going to survive? Or, better even, is anything going to thrive? Because by finding something that thrives here, it can then be sort of determined if this organism is thriving because it's actually eating the viruses. And there were a couple of discoveries. One of them was an extremely common organism known as Paramecium that was seen to have survived and seemed to have consumed some of the viruses, potentially using them as a kind of a snack. And this is somewhat similar to the discovery from a few years ago that discovered another protist capable of doing something similar. And this of course implies that there are quite a lot of different single-cell organisms that might have actually evolved to potentially use viruses, at least to some extent, as a potential source of additional nutrition, especially when conditions are not very friendly. But there was a much more important discovery from this study in regards to Halteria. Even though everything else disappeared, these little guys grew in numbers by approximately 15 times in just 2 days, while at the same time the number of viruses dropped dramatically, which of course at first implied only one thing: these viruses were being eaten by Halteria, and they were using them as a primary source of nutrition. Moreover, according to the scientists, they even grew quite dramatically in size, and certain DNA fragments from the virus were then seen inside Halteria using the fluorescent green dye. You can sort of see it right here as the tiny green spots. But more importantly, they were mostly concentrated inside the feeding vacuoles, or basically kind of like the tiny stomachs where the nutrition was stored, which really only meant one thing: they were definitely eating the viruses, and they were definitely growing and multiplying because of it, making this the only such organism known to us as of 2023, which means that Halteria and chlorovirus have a very unusual predator versus prey relationship that's never been seen before. And although many different organisms were discovered to occasionally eat viruses, so far this is the only one that seems to prefer them. Although funnily enough, the actual entrance for virovore on Wikipedia is still extremely short. This is the only such discovery. But more importantly, there was also an additional discovery on top of this that to some extent affects the modern understanding of what's known as the carbon cycle. Chloroviruses, as the name suggests, prefer to infect various green algae, or basically algae responsible for producing huge amounts of oxygen on the planet, but also obviously fixating a lot of carbon dioxide. And so green algae in this case represents one of the biggest carbon sinks in the oceans and is directly responsible for producing oxygen, but also fixating CO2. More green algae, more oxygen, less CO2. But the presence of these viruses destroys the green algae and thus has the potential to suddenly release a lot of CO2 into the oceans and also obviously lower the oxygen production. And so even though this was just a tiny sample, in reality this represents a huge amount of biomass with the potential of releasing huge amounts of carbon, or vice versa, potentially storing a lot of carbon. But this is where Halteria comes into the picture as well. It definitively plays a role of a kind of a regulator by essentially destroying chloroviruses and allowing more algae to exist. And so, at least in theory, it might have a huge effect on the carbon cycle on the planet, but may also explain a lot of other phenomena we don't understand, such as for example the occasional algae bloom that tends to dramatically increase the production of algae for reasons still not understood. So basically these little guys definitely play a role of regulators of the release of oxygen and the fixation of CO2, but what effect all of this has on the planet is still obviously not known. It probably has some other effects on the food chain in the oceans and the release of a lot of other nutrients from, for example, algae itself. But before the scientists can discover any of this, they first have to confirm that this is exactly what happens in the wild as well. They basically have to observe this in the actual pond or, for example, some kind of an ocean. At the moment, all of this was just done in a lab and in very artificial conditions. So we don't really know if this is how it works out there in nature as well. And so this is definitely a pretty exciting discovery. Although just to clarify, they don't just eat viruses, they also do eat other bacteria, so these are kind of like predators of a typical pond or a typical ocean. But the fact that they seem to prefer these chloroviruses is a really important discovery in helping us understand what happens on a microscopic scale when it comes to the carbon cycle. And so chances are we're going to be talking about this organism again sometime in the future. In the last few years, we've heard a lot of different stories and a lot of different announcements in regards to quantum computing. The idea that's supposed to completely revolutionize the computers as we know today. But the idea that even today is still kind of struggling to find any practical use or to even create something useful outside of a complex machine that's able to solve just a handful of complex mathematical equations. In other words, despite all of the hype and all of the announcements, at least for now, quantum computing is still just an experiment that seems to be kind of cool and seems to work, but does not really have a very good practical use. But when it comes to innovative computing ideas, or essentially completely new types of computers, quantum computing is not the only contender. On an almost completely opposite scale, we have the idea of biological computers. Or essentially the idea behind biochemistry driving most of the mathematical computations. And though it might sound kind of far-fetched, it's actually been in existence for over 3 decades, with some of the recent announcements basically taking us to a completely new level. As we're going to discuss today, based on this recent study, we actually could be entering a new era of what scientists refer to as DNA-based programming, or basically DNA computers. And though by itself, once again, it might sound kind of far-fetched, it really isn't. I mean, just look in the mirror. You and I are some of the most complex biochemical computers / robots in existence on planet Earth. And so this is definitely physically possible. But the question is, of course, can we control it? Can we turn it into an actual computer? And can this become practical? And honestly, the answers to all of these questions, at least for now, are it's not clear. But this is exactly what these new studies are about. But as I mentioned, this is not a new concept. As a matter of fact, the original experiment that tried to show that this is possible was back in 1994. Leonard Edelman, that you see right here, presented the first prototype of a DNA computer known as TT100, where he basically used a tiny tube filled with 100 microliters of DNA to solve one of the basic mathematical problems. Hamiltonian path, a type of a path optimization problem meant to establish connections for the shortest path possible. It was successful back in 1994. And this was actually the first application of DNA to mathematical problems. A very similar approach in 2002 was able to solve even more mathematical problems, including 3 well-known SAT problems that actually had 20 mathematical variables in them. And so even by early 2000s, these very simple DNA biochemical procedures were able to physically solve mathematical problems that we normally use computers for. Although obviously here, it was all based on biochemistry and chemical reactions, and molecular biology as the main hardware. But the main problem here was, of course, twofold. First, this was relatively slow. A lot of these reactions would often take hours or maybe even days to complete, meaning that to solve the problem you do require a lot more time. Which is practically the opposite approach from quantum computing, where usually the results are almost instant. And the other main problem here was the analysis. It obviously takes a long while to get the results. There is no direct biological screen to tell you right away what actually happened. Or at least not yet. But this was a good start. It looked like we could use biological systems for very similar mathematical calculations to a typical computer. But it wasn't really until the last few years that we started seeing some major advances.
mostly because of major advances in genetic modification, including of course the famous CRISPR. One of the first intriguing announcements back in 2016 was the breakthrough in information storage. A team of scientists was able to encode 4 separate images into strings of DNA by converting digital data into 4 basic elements of DNA molecules: adenine, guanine, cytosine, and thymine. And so essentially they were able to store huge amounts of data in a tiny amount of DNA present inside this pipette, proving that DNA can serve as an ultra-high density storage for all sorts of data. But once again, the encoding and decoding was not very fast. Here the process took approximately 100 hours. But that's at first. Eventually scientists realized that you can actually do tiny strands all at once in different locations. And this dramatically opens up the doors for DNA computing. By providing correct instructions to different sequences, you can dramatically speed up performance by millions or even billions of times, essentially creating a biological parallel computer, dramatically increasing the performance of these mathematical solutions by using just a tiny fraction of energy of a typical classical computer, or more importantly, a quantum computer. Here you have a tiny setup that's essentially just one pipette, with all of this happening all at once, all of this running by itself with just a tiny amounts of energy, and producing relatively accurate results. And so in the last few years, this definitely formed really exciting opportunities. But now we have a very big breakthrough. The world's first DIC, DNA Integrated Circuit. Something that's supposed to have a much more general purpose, and that's not just going to be useful for specific mathematical solutions. And something that's essentially a liquid DNA computer. A directly programmable biological device that allows us to apply various algorithms similar to a classical computer, and most importantly, allows for scalability and thus increased speed, with the theory already presenting a possibility for up to 100 billion different circuits running in a similar way to a typical computer. And so in this case, if you look at the picture, it resembles a typical gate array, a type of a specific integrated circuit that was extremely popular in the 80s and in the 90s for a lot of early computers and early electronic devices. Here though, each of these short DNA segments essentially create larger structures able to use molecular instructions in the same way that electricity controls the circuit. With the first experiment already showing that you can solve quadratic equations through a very simple chemical reaction that transforms the circuit inside. With one of the most important achievements here being the fact that there's really no loss of signal no matter how big the structure is. And so here, no signal attenuation is required at all because these chemical reactions do not produce any kind of a loss. And so even by producing a large enough machine, it's possible to create something that's 100% efficient. And though obviously this is still just a proof of concept, and we're not going to be able to produce anything like this just yet, the ability to create these large DNA networks that are able to be very efficient, very small in size, and require almost no energy, if scaled to a large enough size, in theory, can definitely outcompete classical computers. But this is just the first step. Here they only used 30 different logic gates and only 500 DNA strands. But even this really tiny system was able to solve quadratic equations, providing the correct answer. And so there's quite a lot of potential here. Assuming this can be scaled up and assuming this can be actually developed into something that's easy to implement, at least in theory, it could lead to some innovations in biological computers, with one of the biggest potential being biological sciences. Here the implication can be that you could use these computers to directly classify various viral or bacterial pathogens by running them through this biological computer without the use of anything else. Although here, once again, we have to be a little bit careful. Just like with a lot of other grandiose announcements in the last few years, for all we know, maybe this is as far as it gets. We still don't really know if this is going to be possible practically, or if anyone is going to find a way to scale these computers to much larger proportions, or more importantly, if they're actually going to work at all. For example, what if, just like the biological tissue, all of these computers require specific temperatures, specific pressures, and very specific pH? Or what if your biological computer gets sick? What happens then? Is it going to provide you with completely wrong results, or is it just going to die? So still definitely a lot of questions to answer, but a pretty exciting new discovery. We'll of course see where all of this goes in the next few years. When it comes to search for extraterrestrial intelligence, or really any kind of life out there, one of the more important things we have to understand is actually how vision works. Or I guess more specifically, what types of vision exist and how it evolved over time. And for two main reasons. First, we know vision is very important for humans in both our perception of the world and in how we analyze everything around us. And second, because many different complex animals on our planet either rely on vision for survival or rely on displaying certain colors in order to prevent being eaten. Not to mention that it's also believed vision was probably the main reason such a huge complexity of life evolved over time. And furthermore, if some kind of intelligent life exists out there, on a different planet in a different star system, it's most likely going to rely on vision as well, in order to learn about everything around it, and in order to then learn about the rest of the universe. And that's because all of this information from various galaxies and various stars is really only detectable in certain electromagnetic frequencies, implying that any extraterrestrial intelligence would very likely have a form of vision able to receive this information. This of course means we have to understand vision and its evolution, as well as understand various complexity that it comes with. And so, hello wonderful person, this is Anton, and in this video we're going to be discussing one of these more unusual discoveries made very recently in regards to a very unusual type of vision that seems to be present in this fish. And in a separate video that's going to be coming out sometimes in the future, we're going to be discussing other unusual types of vision in a lot of other types of complex life. Mostly because this topic is actually really fascinating and there's quite a lot to discuss. But let's start with this recent discovery. This is actually in regards to a very common fish known as hogfish, also known as Lachnolimus maximus, a species of wrasse that's usually found in western Atlantic Ocean, very often around Nova Scotia, Gulf of Mexico, and in some parts of South America. And the only reason they're named hogfish is because of their unusual long snout, and because they use this snout to dig in the ground similar to a typical hog or a pig. And they do this to catch various crustaceans living in the sands. And generally, this is a pretty large fish. It can reach up to about 90 centimeters, or about 11 kilograms in weight, 36 inches, 24 pounds. But one thing that makes this fish somewhat unusual is the fact that when it's young, it's completely different from when it's old. They generally start with somewhat gray or brown color, making them look somewhat simple and not particularly interesting, but they also start with both male and female sex organs. They're basically hermaphrodites. And this is kind of common in some types of wrasse, but not super common in a lot of other fish. And they actually change their sex several times. They first become female, they then become male, with the first change occurring around 3 years of age, when they're actually really small in size. But because they can live up to 11 years, after changing into males, they can usually survive for a very long time. And when they do this sex change, they also transform their skin. They basically learn how to change color. And most likely for the same reason as octopuses, camouflage. Mostly to help the fish blend in with the coral, or to maybe sometimes use in various mating rituals. But by itself, this discovery of color change in fish, I guess, though surprising, is not particularly super exciting. What is exciting is the discovery of what's underneath the skin, something that was recently reported in this study you can find in a description. And so the authors behind the study, specifically Lori and Schweikart, was able to take extremely accurate images of the skin using transmission electron micrograph, in a process discovering intriguing structures underneath the skin itself. Now, first of all, to change color, like in a lot of other animals like squid, cuttlefish, and octopus, these fish use chromatophores, types of unique cells able to change color over time. This is actually something that's used in chameleons as well, but turns out these pigment cells bundle with something underneath them as well. They seem to contain molecules called opsins right underneath everything as well. And in every organism containing vision, opsins are usually responsible for sight, suggesting that these newly discovered opsin cells are very likely sensitive to certain types of blue wavelengths that can pass through upper skin, in essence allowing this fish to be able to see with its skin. Although I guess using the word see or vision might not really be appropriate here. A lot of these opsin cells seem to be independent of any neurons and do not connect with the brain. They seem to act completely independently. but essentially creating their own network, a kind of a distributed site. But that can also potentially allow the fish to sort of sense the environment around themselves by perceiving slight changes in skin color. So for example, an approaching predator might cast a shadow, changing the response on the skin, something that this fish would most likely be able to sense, thus hiding somewhere in a coral. But because this visual network is independent of the brain, it very likely acts extremely fast, resembling a kind of an instinct. Although here scientists do state that it's not clear right now if this is only for light detection, or if it actually does form some kind of a three-dimensional image that the fish can then sense. But today we're pretty certain that octopuses and cuttlefish are able to do all of this as well. They also contain opsins in their skin, and it's usually a part of a sensing network responsible for sensing the surroundings. It's very common in cuttlefish and of course squid. If you've ever seen a cuttlefish in real life, you probably noticed that they seem to go through a variety of colors for a lot of different reasons. Sometimes communication, sometimes signaling other cuttlefish, but more importantly, they also are able to sense what's behind them or what's above them by sensing minute changes in shadows and minute changes in light reflecting on their skin. So when it comes to being able to see with your skin, these guys seem to be the best. Which might be very different for octopuses. They seem to mostly use their chromophores for camouflage, not necessarily communication. And so octopuses and cuttlefish seem to have evolved different types of skin vision. And we know that for a fact because their opsins seem to be present in different structures. In octopuses it's found in cilia, in squid and cuttlefish they seem to live inside chromatophore cells. But all this is a result of approximately 300 million years of evolution because that's when octopuses and cuttlefish split into two separate species. Nevertheless, understanding these different types of vision and how all of this works in a lot of different animals right here on planet Earth is somewhat important for a lot of astrobiological studies as well. Mostly because we would assume that something similar would maybe evolve there as well. Although I guess since most of the stars out there are usually red dwarfs, life living around those stars might be sensitive to very different frequencies of light. For example, infrared light is extremely common around red dwarfs. But for now at least, these are all just assumptions. We're not going to be able to answer any of these questions until years and even decades in the future, and until potential missions to objects like Europa, one of the moons of Jupiter, that might contain some kind of complex life underneath. At least this is one of the best potential candidates. So basically studying vision right here on Earth is a really important factor in trying to make assumptions about extraterrestrial intelligence. Most of the videos on the channel that many of you are used to are about planets, stars, about the universe out there. But today we're going to come back to Earth and talk about something else incredible pretty much right under our feet. Something that as always was discovered completely unexpectedly, somewhat by accident, very close to Boston in the US. And with a sample collected back in 2019. And here, when the scientific team put these samples under extremely powerful transmission electron microscopes, the ones that usually use the electron beams in order to magnify an object, they discovered an incredible, unexpected biome that we've never seen before. An entire collection of incredible looking viruses, some even possessing what seems to be basically arms, some coming in very unusual, never before seen shapes, and some even having something that resembles hair, with many of these viruses being extremely complex and actually being some of the largest viruses we've ever seen. In essence, possibly taking us a tiny step closer, first of all in trying to figure out exactly what viruses actually are. I mean, are they life? Are they something in between? Are they just their own thing that defy explanation? And second of all, taking us a step closer in explaining the origin of life. So, hello wonderful person, this is Anton. Let's come back to planet Earth and discuss amazing structural diversity of giant virus-like particles in forest soil. Because in this case, it's not even certain exactly what these are. I mean, technically I guess we think it's viruses, but they seem to be very complex. And some of them do seem to possess structures and things that no other virus is known to possess. And so let's discuss some of these details. But I guess let's start with the obvious. The things that we already know from previous discoveries, some of which you might find in a description. There's actually a family of viruses known as Nucleocytoviricota Yeah, you try saying that 5 times fast. That's basically known as the group of viruses with extreme complexity and possessing a lot of complex properties that no other virus seems to have. For example, unlike other viruses, especially viruses with very specific mechanisms when it comes to replication, this unusual group of viruses seems to be able to replicate in different ways and even in different parts of the host cell. On top of this, many of them are giant in size. The largest found so far is approximately 1500 nanometers, or about 10 to 15 times larger than a typical cold virus, and at least 3 times larger than viruses we're discussing today. Intriguingly, this virus was actually found in ancient deposits in Siberia from ice that was about 30,000 years old. And intriguingly, even after 30,000 years, it was still able to infect amoeba or its primary host. And so a lot of these marine viruses that often use amoeba or even algae for reproduction become extremely complex and somewhat large in size. But what's unusual for these types of viruses is the fact that even their genome is way more complex than any other virus known to us. It actually involves different types of genes involved in DNA repair, replication, transcription, translation, and all of the necessary processes to maintain DNA that's usually common in a typical cell, but is never seen in viruses. Even when it comes to the size of the genome, a typical giant virus will have anywhere from about 500 to even 1200 different proteins that it's able to code using genes. If you actually watched a previous video on the synthetic cell that scientists were able to create by removing unnecessary genes, that's basically 3 times higher than an actual biological cell. So here we have a virus that seems to have more genes than some of these simple cells. And so by itself, that phylum of viruses has already presented quite a lot of different mysteries. But more importantly, presented a lot of features that we generally associate with actual life. So to some extent, it presented a potential explanation or a potential connection between living cells and ancient viruses. But the results from this study take all of this complexity to a completely new level. Here we discover incredible diversity of these unusual giant viruses, which seem to reveal extraordinary shapes and unusual features that we've never seen before anywhere and that are still kind of difficult to explain. In this case, even capturing cells in the process of being infected by viruses, which is an absolutely incredible image. Here's actually another cell with a slightly different virus. In this case, I believe this is a myovirus entering a cell by basically drilling inside of it. And here's a slightly different virus doing the same to a slightly different cell. And seeing something like this in real life is absolutely mind-blowing. This physically shows us how viruses invade a cell. I mean, obviously in this case, this is all frozen, so it's no longer active, but it got captured right before this virus was able to infect the cell. And intriguingly, quite a lot of these large viruses exhibited a lot of beautiful geometric shapes. In this case, this is known as the icosahedral capsid, with all of them produced in a very similar way. A result of a single amino acid that essentially takes a part of a cell, a host cell, and creates a kind of a three-dimensional origami out of it. But in this case, the size itself actually depends on a single mutation inside the amino acid, which is why some of these cells are able to grow to tremendous sizes, some of which you can see right here. But I guess more intriguingly, many of them do seem to contain additional features. For example, this one they decided to name a turtle, I guess because it sort of resembles a sea turtle. But this is at least 400 nanometers across, making this one of the larger such shapes. Then we have this unusual formation that they now named Gorgon, basically named after Medusa, mostly because of the protrusions resembling the snakes around Medusa's head. And as you can see, this one is also pretty large in size. These unusual protrusions, whose function is obviously unknown, seem to be at least 1000 nanometers across. Then we have these very strange star-like formations, which in this case the researchers named the Christmas star. Exactly how this is formed and what function any of this has is obviously unknown. Then we have that weird hairy one with really strange formations on the surface, in essence showing a very large diversity of capsid fibers. Very short, thin fibers on the surface of the geometrical capsid, whose purpose is obviously once again unknown. Now it's most likely used for some sort of an attachment, or to possibly help invade the cell, but that's just guesswork. Then there are these two that they think look like supernova, which I guess is kind of true. and so they named them supernova. These viruses seem to contain very similar fibers, but in this case entangled across the capsid, creating an additional thick layer. But they also seem to possess other tendrils that extend beyond. We also have some more unusual tubular shapes. In this case they actually call them the plumber, but also discovering unusual triangular shapes we've never seen before in any virus phylum previously observed. Which of course means that this is possibly a completely new discovery. And honestly, it's just worth going through some of these pictures because a lot of them are absolutely mind-blowing. It's insane how far the technology has come when it comes to electron microscopy. We can now take pictures, very accurate pictures, of what seems to be some of the smallest things out there, viruses, with the images themselves being relatively high quality and very detailed. But obviously this is just morphological observations for now, just showing us how extremely diverse all of this is. The true function of any of this is not really going to be known to us for many years. And what's even more mind-blowing is the fact that this was discovered in a typical US forest in just a few hundred grams of soil, with just a ridiculous diversity discovered after just a few images. A unique hidden world right under our feet. Super impressive, incredible work, and incredible images. But I guess once we actually figure out what's going on here, or once someone discovers what some of these viruses are, we're probably going to come back and talk more about this, because at the moment there is really no explanation. These viruses are definitely important for a lot of different ecosystems, and as you might have learned from some of the previous videos, quite a lot of viruses are also important for our own life. As a matter of fact, as I mentioned before, approximately 7% of your DNA is viruses. But the incredible genetic diversity of these types of viruses is of course of particular interest because they might help us answer questions on the origins of life. But until we get closer to these answers, or until something else really good is discovered here, that's pretty much it. So in the last few months there's been a tremendous amount of research and various discoveries in regards to the human brain. So many discoveries, as a matter of fact, that I guess I'm going to have to divide this video into several parts. And in this first part, we're going to discuss recent announcements and recent discoveries coming out of NEMO, the Neuroscience Multi-Omic Data Archive, essentially representing the largest collection of a lot of different data, or more like huge amounts of data, in regards to the human brain. And extremely recently, at least 24 different papers were published using all of this data, in the process of revealing some of the strangest things about the human brain we've ever discovered. Also creating the largest human brain atlas ever, specifically focusing on discovering different types of brain cells and what they possibly do inside our brain. Here are some of the images of some of them. But these 24 papers represent the first ever whole human brain atlas focusing on individual cell types and molecular interaction with the rest of the cells. And what's absolutely insane here is that the scientists identified over 3,000 different types of cells inside our brains. All of them responsible for different types of functions, and most of them unique to specific areas of the brain. And so we don't just have neurons and support cells like glial cells, we actually have 3,000 or more than 3,000 different cells already identified inside the brain. With this also being the first collaboration trying to focus on differences between human and non-human brains, with the emphasis being molecular interaction, not just the structure. And so there's definitely been some incredible discoveries in just the last few months. And so, hello wonderful person, this is Anton. Let's discuss our brain, let's find out what was discovered, at least to some extent, because there's just a lot of stuff discovered here, and talk about some of the implications. First though, what exactly was this collaboration? It started in 2017, and it basically focused on 5 main themes, trying to create the overall atlas of the brain, comparing this to various brains like monkey brains, also comparing this to non-primates like mice, trying to figure out how various cells develop, but then specifically focusing on individual cells depending on the structure of the brain. And because we know the brain contains at least 86 billion cells with at least as many support cells, this was obviously no easy task. In terms of the actual number, that's just a little bit less than the number of stars in the Milky Way galaxy. Yeah, that's a lot of cells. And not surprisingly, many of these cells turn out to be very different. But what was surprising was the location of the complexity. Turns out that the parts of the brain we normally refer to as more complex, so basically cortex, where the thinking happens, were really not that complex in terms of the actual cell structure. And the most complexity was discovered in the older, deeper brain, with the most complex of them all being the brainstem, an extremely understudied structure that connects the brain with the spinal cord. And though it only makes up about 2.5% of the total brain weight, it does have a lot of different functions it's responsible for. It regulates the heart, the breathing, the sleep cycle, and also serves as a kind of a interconnection between the brain and the rest of the body. And so this tiny structure turned out to be the most complex when it comes to the actual cells present inside the brain. In comparison, our visual cortex, the part of the brain we use for seeing, is way more specialized and much more distinct compared to other structures. It's even more specialized than in other animals, for example mice. And that's probably because humans and a lot of other primates mostly rely on vision to navigate the world. Despite of this though, there are a lot of cells in this case that seem to be specialized and seem to be extremely specific in order to help us visually understand the world around us. But one big question here is, of course, how does this actually compare from person to person? Are there a lot of individual differences? And turns out that there definitely are. Every individual seems to have a proportion of certain cells different from another person. And more specifically, the genes switched on in various cells to control them seems to be different from person to person as well. And so even on cellular and genetic level, individual human brains are totally unique. And since we're talking about 86 billion cells in this case, yeah, we're definitely individual snowflakes. But in terms of the overall structure and architecture, we don't just share it across the species, we even share brain structures with various evolutionary cousins. And so in terms of the actual physical structure, the human brain does appear somewhat similar to, for example, a chimp. But it starts to become extremely different when it comes to genes controlling the connections between neurons and the formations of various circuits inside the brain. Here things become extremely different, because the human brain uses different genes to create these circuits in slightly different ways compared to everyone else. And more interestingly, even the exactly same gene in the same type of a cell can have an entirely different effect in a different part of the brain. So basically you require different genes, different types of neurons in different parts of the brain, adding to the already complex interaction. And one of the main reasons for this types of research is to, of course, try to understand various types of disorders we humans experience in our brains. And it turns out that a lot of human brain disorders are basically unique to humans. They don't occur in any other animals, including our cousins primates. And so studying these cells and trying to understand them is extremely important. And even in these first papers, there's already been a link discovered between various types of brain cells and a lot of different neuropsychiatric disorders. Things like depression, schizophrenia, and bipolar disorder. One of the studies, for example, focused on what's known as microglia. You can see these cells marked in green. They're normally responsible for cleaning everything and for removing damaged cells, but turns out that certain genetic components seem to act on these cells, dramatically increasing the risk of things like Alzheimer's, to some extent suggesting that the basis for many psychiatric disorders could be entirely genetic and molecular in nature, but also implying that it's super complex, way more complex than anyone ever thought. Another finding was about this unusual layer discovered by accident. It's now referred to as SLIM, subarachnoid lymphatic-like membrane that basically appears as a kind of a barrier, potentially serving as part of the immune system protecting the brain. But because the brain itself does not have an actual immune response, unlike the rest of the body in this case, it has what's known as immune privilege, a part of the body that doesn't produce immune response, so for example things like eyes. In this case, by discovering this unusual layer with several different immune cells such as myeloid cells and macrophages in it, suggests that this organ seems to basically watch the brain by controlling the flow of cerebrospinal fluid. I guess a kind of a sewage control with liquid going in and out of the brain. And so by transporting and removing the waste from the brain, it also supports the immune defense, preventing anything dangerous from getting inside the brain. And so the discovery of this unusual layer relatively recently was definitely quite surprising as well, but helps us understand a little bit more about the brain and how it maintains its health. There was also an intriguing discovery in regards to what's known as axon fibers. In general, we know that all animals will contain these axons, which are responsible for carrying electrical impulses, and previously were always believed to be a kind of a branch-like formation that forms from inside the neuronal cell. But this new study discovered something entirely different. First of all, it turns out that they actually also originate from dendrites, a slightly different branch that you see right there, that are very often responsible for connecting neurons together. It wasn't previously thought to be possible, but turns out that it is. But that's not the big discovery here. The big discovery came from studying this in a lot of different animals. And turns out that these unusual axon-carrying dendrites, or basically axons that were forming not from neuronal cells but from dendrite connections, are much more common in non-primates like cats and pigs. Which by itself is a very interesting and somewhat unusual discovery that is somewhat difficult to explain. It basically means that in, for example, humans, we're most likely to have impulses and signals come directly from neurons via axons toward another cell. But in, for example, cats or pigs, this can also happen from dendrites without the use of neuron. And because neurons are normally a kind of a gatekeeper when it comes to deciding which signals get passed and which don't, and these dendrite-carrying axons seem to have the ability to kind of ignore the neuron and basically pass the signal anyway, it suggests that the signals in brains of other animals are passed around in a somewhat different way. With the additional discovery in this case also suggesting that the animal domestication did not affect the animal brains at all. Both pigs, which are domesticated, and boars, which are wild, seem to have exactly the same structure when it comes to axons. And so that's one of the differences we've discovered about human brain versus non-human brain, but exactly what it means yet is still unknown. Another discovery comes from something else. It comes from the study of the shape of the brain and how it affects the skull itself. In this case, studying individual brain cavities in order to get approximation of the brain's shape. And so by looking at changes in the shape of the brain as the animal grows up, so basically as the head itself changes in shape with time, scientists discovered that animals like apes, so chimps and gorillas, seem to have very similar integration between various brain parts compared to humans. At least until they reach adolescence. In other words, their brain seems to mature in a similar way and seems to grow in a similar way for the first few years of their life. But it seems to change dramatically once they become adults. The brain growth sort of solidifies, and various brain parts no longer change their integration, basically stopping their advancement and reaching a kind of a static state. I guess for the lack of better words, they kind of stop learning. Turns out though, in Homo sapiens, which is us, and our extinct cousins Neanderthals, things are very different. Our brain seems to never grow up. It seems to experience continuous integration even into adulthood, with the brain shape and the skull shape changing even decades into human or Neanderthal life. Which of course implies that we don't just learn until we become adolescent like chimps. Our brain seems to continuously learn and evolve even past adulthood. Which shouldn't really come as a surprise because, you're watching this video and you're learning right now. And most of you are very likely adults, at least according to my YouTube statistics. But what is surprising is that that's not the same for chimps. They seem to stop the learning process pretty early on. They obviously still learn a little bit, just not as dramatically as we do. And so this actually suggests that it's not that the larger brain that seems to define intelligence, it's really this overall integration inside the brain that seems to continue for a very long time. And more importantly, it also implies that our cousins Neanderthals were extremely similar to us. Not some brutes that didn't know what they were doing, they were extremely intelligent. There's actually a lot of evidence about this, and you can learn more about this in some of the previous videos. But this is of course just one of many things that make us human. And this is just a study based on CT scans of various skulls in order to understand how the skull and the brain evolves as the species grows into adulthood. We're going to actually discuss a lot of these discoveries separately in a video involving genetics and molecular studies that basically help us understand why humans are human. If you don't want to miss that video, make sure to subscribe. But there was one more discovery from a few months ago that once again highlights how human brain seems to be different from other animals. And once again, it doesn't necessarily make sense. This was a study based on hippocampus, which usually plays a role in memory, but also seems to play a role in visual imagination and our ability to construct mental images. It also plays a role in navigation, usually by helping us form mental imagery and by connecting the visual cortex with various parts of the brain that make decisions. And turns out that once again, it seems to be different in humans. Different from not just other animals, but even from primates. Animals like chimps and gorillas. For some reason, human hippocampus has a much smaller connection between the frontal area of the brain, where we do most of the thinking, and much greater connections with the visual areas. In other words, even though we kind of expect our complex human brains to have more connections between the memory center and the part of the brain where we do the thinking, so the prefrontal cortex, in reality, the connections seem to be most prominent with the visual areas. And that's probably because of some kind of a human-specific memory that relates to visualization, imagining things, and visual cortex, which to some extent might potentially finally explain why humans compared to other animals are so creative. I mean, the idea of art itself, our ability to paint, to create a lot of different imagery, to create sculptures, potentially comes from this very small but very important difference discovered only in humans. But when it comes to the idea of memory formation, it's still super complex for us to understand. Even recently, in one of these studies I mentioned previously, and the one that you can obviously find in the description, scientists once again discovered a new type of a brain cell that they now call glutamatergic astrocyte. It seems to be not really a neuron, not really an astrocyte, something entirely different, but something that seems to play a role in active memory formation, because when they disrupted these cells in mice, those mice started to have major memory problems. But even the regular astrocytes, the cells that you see presented in green in this image, which were always believed to be more or less passive, and basically responsible for the physical protection of various cells, turn out to be a lot more active than anyone believed. They actively respond to various signals and seem to be directly responsible for helping the brain recover from various types of stress. They do so through a very complex chemical signaling system that was just discovered in one of these papers. And so all of this of course suggests that our brain is ridiculously complex. Way more complex than we even thought a few years ago. Even compared to that previous video from I think 2 years ago that you can find in the description. And a lot of these discoveries seem to be directly unique to humans, which of course is the big question here. So what exactly makes us so unique, and what makes human intelligence so different from anything else on the planet? And we'll be discussing all of that in the next part that's going to feature a lot of other papers from the last few months. There's been a tremendous amount of discoveries that I just can't cover in a single video. A lot of these discoveries are in regards to different cells that we never knew existed. Some of them are about genetics, only in humans, and some of them are in regards to how the brain develops over time. And so we'll definitely discuss this in the future, in a video that's going to be coming out, possibly in the next few weeks. Although here I guess I also have to mention the limitations from these studies. So far, most of this has been done by using tissue from various donors, people who passed away, or possibly tissues donated as a result of a surgery, such as for example a brain surgery involving epilepsy. And so in that sense, it may still not necessarily represent a living, healthy human brain. However, because many of these studies were done using RNA analysis and by using various genetic switches, for the most part the discoveries seem to be more or less accurate. But it's still important to mention these limitations. So approximately 10 years ago, I watched a few YouTube videos of someone playing The Last of Us. Actually, I think it was Markiplier. And I was so impressed that I decided to try to get my hands on the PlayStation 3. And I literally spent 2 weeks playing the game because it was just so good. And not even the gameplay, just the premise itself, the story, the idea behind it, and even the portrayal of these unusual fungi zombie, or basically clickers. And you can probably imagine my excitement when I found out that they're also making a TV show. Oh yeah, there's also part 2 of the game, but I'm really behind the schedule on my games, so I haven't played that just yet. Anyway, hello wonderful person, this is Anton. So today we're going to be talking about the idea behind The Last of Us. The idea behind the fungal infection. And basically answering the question of, can this actually happen? Is there a possibility for an actual fungal infection that can infect humans, potentially turning us into zombies, or exhibiting other effects, possibly deadly to humans? And even though this might sound preposterous, you might already know where I'm going with this, because this right here is kind of based on the real thing in nature. Not in humans obviously, not even in complex mammals, but in insects. And if you know anything about the original game The Last of Us, it was actually inspired by the episode from BBC. The episode that you see right here, as always narrated by David Attenborough. And this actually shows us what happens to some of the ants infected by certain fungi as they literally turn into a kind of a zombie, with the fungus then starting to grow, usually out of their head and a few other body parts, and eventually releasing spores, potentially infecting other ants or other insects depending on the fungus, with the family of these fungi known as the cordyceps. Although for these ants, the more specific name is Ophiocordyceps, a very specific fungus that parasitizes specific ants, with a single fungus only able to control a single species of ant. And because there are several fungal pathogens known to us that can actually change the behavior of the animal they infect, this idea might not really be that far-fetched after all. So let's discuss this from the perspective of science. And by the way, there are going to be no spoilers about the TV show or even the video game. I'm just going to be covering basic concepts. And a lot of these discoveries are somewhat recent. As a matter of fact, some of the first papers on this fungus and its effects on ants only started to come out about a decade ago, around the same time as the video game. Here's one paper you can find in the description. And in this paper, the scientists explored how Ophiocordyceps managed to evolve various types of compounds to influence various types of behaviors in specific types of ants. And what's intriguing is that they actually evolved really specific components that seem to do very specific things, usually divided into 5 separate steps. The first step seems to be relatively similar. The ant picks up some of the fungus that gets stuck on its skin, and the fungus slowly finds a way inside the ant's body by using various enzymes to break through its skin. The second part is a little bit more specific. It involves direct manipulation of the ant's behavior. The fungus does not actually infect the brain, but seems to develop a kind of a colony very close to the brain itself, which then starts to emit very specific metabolites that almost instantly start to affect the ant. And in almost all cases, in almost all species, the first thing that happens to the ant is a type of a convulsion, where the ant suddenly loses footing and basically falls to the ground. This happens in almost every case. Later on, the fungus kind of spreads in different ways. Some fungal spores end up in the mandible of the ants or the mouth, others end up in the muscles, with others still being close to the brain. And they all start to emit different metabolites, which start to affect pretty much everything the ant does. And following the convulsion, once the ant gets up, it generally starts to find a way to get above the forest floor. And what's super intriguing is that even the height here seems to be really specific. The scientists discovered that it seems to prefer the height of about 26 centimeters above the floor, and seems to always prefer the northern side. It also prefers 95% humidity and a temperature of 20 to 30 degrees Celsius. And if it finds these conditions, it initiates the last step, known as the death grip, seen in the video by the BBC. It grabs onto the stem, bites really hard, way harder than usual, with the mandibles staying fixed pretty much forever. And as soon as this happens, the fungi literally kill the ant, using the rest of its body for their own sustenance. With all of this done just so that the spores can easily propagate through the air and potentially infect other ants or move elsewhere. So essentially this is a way that this particular fungus moves across the forest. So it's actually both the mobility strategy as well as the reproductive strategy. And we know that this strategy existed for at least 130 million years. The scientists literally discovered the signs of those mandibles biting into various leaves and various stems in a lot of different types of amber, consisting of both ants and ancient plants. So this is a super ancient relationship. It's been around for a very long time. And that's a very important part of the answer to the original question. Can this happen to us? Or I guess, can this happen to us? Well, no, not really. Or at least very unlikely. If this could happen to us, or if this could happen to mammals or really any more complex animals, it would have already happened in the last 130 million years. The ants and the fungi have been doing this for so long that if the strategy was successful elsewhere, it would have already been applied to some other animal. As a matter of fact, fungi are also really dangerous to frogs, and they might even cause the extinction of several frogs in the future, but none of the frogs ever turn zombies. They do experience a lot of problems, but none of them are related to either behavior or their brain. And that's of course because ants compared to frogs are just way more simple. The brain of the frog and even the circulatory system is way more complex for anything to control as easily. But ants are really simple. As a matter of fact, this only takes about 4 to maximum 10 days to achieve, which is why it's so common in insects and is unlikely to happen in anything else.
But ironically, this fungus that infects ants even has its own fungus, parasitic fungus, that seems to infect itself. So dangerous fungal infection in simpler species is definitely a lot more common. And so is the human brain just a little bit too complex for any of these fungi to infect us and to control our behavior? Well, actually, not really. As a matter of fact, we know of at least 2 different parasites that do manage this really well, and they are pretty simple as well. One of them is of course the rabies, the virus that actually affects the neural system, very often producing a lot of aggression, and almost always being fatal to humans, but the other one being something that not a lot of people are aware of, but a lot of people already have. Toxoplasma gondii, T. gondii for short. And this parasite, which by the way is going to have its own video, is something that we know for sure changes behavior in humans, changes behavior in a lot of more complex mammals, and as you might already know, or might not know, is actually spread by cats, and a lot of other felids as well. And this weird parasite has already been implicated in things like psychosis, in things like schizophrenia, and even things like risk-taking behavior. As a matter of fact, what makes this particular parasite really intriguing is that when it actually infects rats, it makes those rats less fearful and often makes them approach cats, which then ends up basically being fatal to them. But what effects this has on humans is actually still not really well understood. We know that it definitely has effects, but so far a lot of studies have been kind of inconclusive. Definitely subscribe if you want to learn more, as the video about this is coming soon. And so toxoplasmosis is a really good example of how humans can also be infected by something where our behavior can change. But we're not really talking about any parasite, we are really mostly focusing on fungi. Knowing what we know, can fungi do something similar? Can they actually turn us into these ants? Well, the answer here is still no, very unlikely. For several reasons once again. First reason is that atypical cordyceps only affects one single species of ant. If it infects another ant or another insect, the metabolites it produces usually have almost no effect. And they definitely have no effect on humans whatsoever. As a matter of fact, a lot of different Asian cultures even use these unusual fungi for a lot of different treatments in Eastern medicine. Creepy, but potentially beneficial. On top of this, in order to manipulate a human, the fungus would have to go inside our brains. But none of these fungi ever do. As a matter of fact, they always stay outside of the brain and simply produce metabolites. These metabolites would not be able to cross the blood-brain barrier inside human brains. So once again, probably unlikely. On top of this, humans have immune system, and our immune system would take care of any fungi inside our blood. Now in humans, we generally can get fungal infection on our skin, for example athlete's foot, but if it ever gets inside blood, the immune system takes care of it pretty quickly. We also breathe in a lot of different fungi as well, including different modes, and most of them do not do anything to us. Some of them, however, are kind of dangerous. Coccidioidomyosis, also known as the valley fever, is one such example. But generally, even when you inhale a fungus, it sort of dies because our bodies are pretty warm. As a matter of fact, one of the reasons why ants and amphibians get infected by fungi really easily, but not humans, is because of our body temperatures. Warm-blooded animals do not really suffer from fungal infections as much. This paper right here expands on this in more detail. And so our body temperatures also represent a huge barrier for a typical fungus infection that would try to control our brains. And so is it then a bit of a science fiction? Is it kind of impossible? Well, this is here where the answer gets a little bit more murky. We don't really know. As a matter of fact, we know that some fungi can actually manipulate our perception or even to some extent our behavior. For example, the famous magic mushrooms produce psilocybin that can cause hallucinations and alter human mind to the point where it actually alters our behavior. So at least in theory, fungi can find a way to maybe manipulate our minds and thus make us do things we don't want to do. On top of this, last year, World Health Organization released the first ever list of different fungi potentially dangerous to humans. And that's because in the last few years, there actually has been an increase in fungal infections around the world. Maybe due to the climate change, maybe due to something else. But what's important is that it does actually increase the chance for some kind of a super pathogen, a super fungus that can establish itself in mammals or even humans, possibly becoming extremely dangerous over time and obviously maybe even manipulating our behavior. Although more likely, if such pathogen does occur, it's just going to produce something really dangerous inside our bodies, but not really turn us into zombies. This is still way too complex for any fungus to achieve. And so can a cordyceps infection turn humans into zombies? Well, probably no. But can some kind of a fungus appear out of nowhere, possibly causing major infections around the world, or maybe even causing a new pandemic? Well, that is actually not so impossible. And that's why WHO released this particular paper, and why a lot of scientists are interested in studying this, because it's not in the realm of science fiction at all. Fungi can be dangerous, and many fungi have still not been discovered even today. And so definitely something that the scientists are taking seriously.
Spoiler alert. Turns out, the Y chromosome is just as weird as we always thought it was. Because after decades and decades of Human Genome Project, genetic mapping revealed that the sequence here is just not really what anyone expected. The complete sequence of human Y chromosome revealed that it's just weird. No better word to describe it. But not like alien weird or like, oh my god, biology is wrong weird. More like it just doesn't really make sense in a lot of different ways and in a lot of different structures. But anyway, hello wonderful person, this is Anton, and today we're going to be discussing some of the new discoveries about the Y chromosome, and also discuss another study in regards to animals that have lost their Y chromosome and in some sense are still surviving just fine. And so we're also going to try to answer the question of, are humans losing Y chromosome, and what's going to happen to humans when there is no Y left around? And so let's discuss this in a little bit more detail, but I guess let's start with the obvious. Human Genome Project, even though it's technically finished, is not finished. Despite a lot of different mapping that's already taken place, and despite solid understanding of what's happening in our genes, there are still a lot of secrets left here, and still a lot of unanswered questions, including questions about junk DNA. Now that's something we'll talk about in some of the other videos, but there are still some things here that we still do not understand completely. And one chromosome that was particularly challenging to map turned out to be the Y chromosome, the smallest of them all. And so even though a few years ago in the video in the description I've discussed some of the recent details and some of the recent discoveries about the Human Genome Project, it took a few more years and additional techniques to finally finish everything, including the Y chromosome. The chromosome that, unlike other chromosomes, doesn't actually contain that many genes. Its main purpose is to determine birth sex in humans and other similar mammals. And so if the embryo acquires X and Y chromosomes, it technically becomes male because it contains a trigger known as SRY gene, sex-determining region Y protein that sort of looks like this, that's responsible for switching on so-called male genes on other chromosomes. One of the most important one is known as SOX9 gene, the gene responsible for the development of testes. But this can be activated by other genes in other animals, and so technically this particular protein is not really required, but it seems to be pretty efficient, and that's why Y chromosome became sort of the feature a lot of different mammals acquired. Although none of this seemed to have existed approximately 150 million years ago. Back then, X and Y were just ordinary chromosomes, the way they are today in, for example, birds. Birds don't have the same sex chromosomes as us. And there used to be 2 copies of the Y chromosome that contained approximately 1000 genes as well. But it's believed that around this time, this is when SRY gene evolved, and that's when this protein became extremely efficient at activating sex genes. And at this point, the Y chromosome started to change quite dramatically. Various types of mutations led to a dramatic change of Y chromosomes, making them degenerate really fast, losing approximately 10% of all active genes per million years, basically reducing the number from approximately 1000 active genes to just 27. Here's roughly what it looks like now compared to the X chromosome. And because of this dramatic loss of genes, it was always assumed that eventually we might actually lose Y chromosome completely. I mean, obviously it has the genes required to make sperm, and even some genes critical for life. If you look at the bigger picture, copies of similar genes exist on the X chromosome as well. It just, for the moment, Y seems to do a pretty good job. But one of the main reasons it was so difficult to actually try to read this chromosome, or to try to map it, is the fact that it's basically full of junk DNA. Nevertheless, the new study managed to sequence most of it, uncovering that it tends to contain multiple copies of similar Y genes, including genes responsible for sperm generation and genes responsible for testes. But for some reason, some of these genes occur in very strange loops where the sequence seems to be inverted, or strange loops where genetic accidents can sometimes duplicate or even erase genes. In other words, its structure kind of makes very little sense. It's as if it's meant to be somewhat fragile and somewhat easy to mutate and potentially make mistakes. It also contains an enormous amount of structures that contribute to pretty much nothing, once again junk DNA, where there are quite a lot of highly repetitive sequences that actually seem to be even coming from different viruses or genes that haven't been used for millions of years. Now that's common in the X chromosome too, but particularly common in the Y chromosome. Remember, this has at least 1,000 active genes, this one here only has 27. And then, strangely enough, it also seems to have a really large chunk of glow-in-the-dark genes, different types of structures that tend to bind fluorescent dyes. Why this is so is currently unknown. But then it also contains a somewhat pseudo-autosomal region that's sort of like a remnant from when it used to be similar to the X chromosome, and in some cases is identical to some parts of the X. So when it comes to genetics, it's almost like the Frankenstein's monster of chromosomes. It's a combination of everything all together, but also the one that seems to make the least sense. But one question that a lot of scientists are trying to answer right now is, so what's the future of the Y chromosome? Where is it actually headed? Now, different independent studies on genetics, usually using multicultural samples, have already detected that as you get older, Y chromosomes tend to generally disappear. And more importantly, there is generally a sign of degradation in older bodies. And because of this, there have been actually several speculations that maybe at some point in the next 10 million years, the Y chromosome might be gone completely. Now, this is a somewhat controversial statement and it's not really scientifically proven just yet, but it's still something worth assessing because there are definitely signs of the Y chromosome decreasing in overall size and also decreasing with age. And so the question is, can it completely disappear? And what would happen to the species then? Turns out, it's happened before, to several rodents in the last few million years. And though technically losing Y chromosome may mean the loss of sexual reproduction, it doesn't seem to be the case, at least for a well-known case of amami spiny rats. They seem to have found a way to regress back to what it used to be like approximately 150 million years ago, evolving X chromosomes into new male sex chromosomes. These rats, though kind of rare, are quite common at least on some islands in Japan. And they all seem to possess just one X chromosome inside their body, both males and females. But it wasn't always like this. Genetical studies determined that they used to have Y chromosome, but just like in mammals and just like in humans, it's basically disappeared over time. And specifically approximately 2 million years ago, certain types of duplication inside X chromosome were suddenly able to perform the same role as the Y chromosome in their ability to turn on SOX9 factor, which meant that they no longer required SRY gene that's present on the Y chromosome. As a matter of fact, it's believed that both were used by certain species, but over time, this possibly became redundant. And so it's possible that as the Y chromosome started to shrink, there was maybe a mixed population of males with and without Y that would then compete for resources. But around the same time, there was also a relatively major climate change which dramatically raised the sea levels, reducing the size of the islands and thus resources available. And then for some reason, the Y chromosome rats died out, the X chromosome remained. It could have been just luck, or it could have been something specific about the X chromosome that allowed them to thrive better. And they seem to be doing just fine. Basically, inside their X chromosomes, the male sex will usually have a lot more duplicated regions that tend to boost the activity of the SOX9 gene, which leads to the male development, whereas the female mice will often not have any duplication of certain regions and thus remain as female. So basically it's a really simple adaptation that most likely exists in humans as well. Although maybe it's just not expressed as well yet because our genes are just more complex. Nevertheless, if the Y chromosome one day disappears, this is maybe one way that all of this could progress as well. But when it comes to the bigger question of, is this also our future? The answer is not really as clear. First of all, it still looks like the Y chromosome is doing fine for now. As a matter of fact, most mammals seem to have the Y chromosome, and it's not really disappearing yet. But since the Y chromosome is shrinking with time, it's really just a matter of chance before something might evolve on the X that can basically perform the same roles as the Y chromosome is performing right now. And once that does happen, well yeah, at that point there's no point to have Y, so it's probably going to disappear pretty quickly. Now, if that actually happens or not, that's not a question we can answer. In those rats that I mentioned, it was basically a result of some kind of a pressure from the outside, and that's not really something we have right now as humans. As a matter of fact, most mammals with the Y chromosome may not have similar pressures for quite a long time either. And since SRY gene is so efficient at activating the sex genes, it might still stick around for some time. So, in the end, the answer is, I don't know, nobody knows. It's possible, it might disappear, or it might stick around for some time. What is pretty clear though, based on the recent analysis and the recent mapping of the Y chromosome, is that it's just really weird. So much more unusual than anyone thought, and has so many different unusual features and unusual qualities, including unusual sequences, that currently do not make sense. Which means that we're probably going to be coming back and talking more about all of this once there are additional studies and more discoveries. For now, the mystery of the Y chromosome remains kind of unanswered. It's tiny, it definitely shrunk over time, but it might stick around for some time. Hello wonderful person, this is Anton, and today we're going to start with something a little bit weird. A meatball. But not just any meatball. The Mammoth meatball. The synthetically made meatball that allegedly is made from mammoth meat, which was basically produced as a kind of a sensation piece, or clickbait, for a startup trying to create a really intriguing concept that we refer to as cultivated meat, or animal meat produced in a laboratory. Although to make this a little bit more sensational and to basically have everyone talk about it, they decided to produce something a little bit more extreme. Basically meat from an extinct animal. An animal that has not existed for at least a few thousand years. And so in this video, I wanted to briefly talk a little bit more about this, because despite this being somewhat clickbaity, it is still actually a really exciting concept, and something that potentially might produce new technologies, helping us resolve several major problems on planet Earth. But this concept of artificial meat, or cultured meat, is obviously not new. The mammoth meat is obviously new, but we'll talk more about this in a couple of minutes. First, I briefly wanted to discuss exactly how the scientists created this, and more importantly discuss the history and where all of this is headed. And I guess to begin, well, because this is a meatball, this obviously requires meat. And the technology behind this engineering technique comes from earlier biological discoveries, specifically medical discoveries, in regards to tissue engineering. And specifically a technique that allows us to literally regenerate certain organs, even using the cells from the body of the person that needs that organ. Here's for example a picture of a regenerated human ear, with the main principle being pretty simple. By using various stem cells, it then becomes possible to reprogram these cells to produce something a little bit more specific. For example, we can create a tissue-engineered heart valve like the one you see right here, and it would generally require only 3 components. The reprogrammed stem cells, some kind of a scaffolding in order to actually create the structure, and then a lot of additional biomolecules such as growth factors in order to create healthy, mechanically stable cells. And in theory, it's actually possible to pretty much create any human organ as long as you have the right equipment, and as long as you can create the correct scaffolding to then place those cells on in order to help them grow into a healthy organ. Now these are some of the more complex examples. I don't think this has actually been achieved yet, but simpler stuff is already possible, which obviously makes this a really important breakthrough in the medical field. This is usually referred to as regenerative medicine, with the specific field of tissue engineering as essentially the biggest part of all of this. But the foundation of all of this is from what's known as cellular agriculture, or basically farming various cells, reproducing them to really huge numbers, and then using them for some kind of a purpose. Now previously this was mostly used for medical purposes, but a few decades ago, back in 1990, FDA officially approved genetically engineered rennet, making it the first genetically engineered product specifically meant for the production of food. And only a decade later, in 2004, we already had the first startup, in this case the one known as New Harvest, essentially trying to figure out how we can use cellular agriculture, okay I'm gonna try this again, cellular agriculture, super hard word to pronounce, trying to reduce the amount of animals we use for meat by replacing the meat from those animals with what we now refer to as cultured meat, animal products that were actually created in a lab. With one of the more successful stories in the last decade being the one from 2013, the rarest, most expensive burger ever. Created from the meat, grown from a tissue, where approximately 20,000 very thin strands of muscle were placed next to each other to try to recreate the texture. But in this case, this cost over $300,000. and also took approximately 2 years to produce. And so back then this was more of a novelty, and thus most people kind of forgot about this over the years. But the technology did not stop. It kept evolving, the techniques kept getting better, and the scientists found new ways to create various types of artificial meat much cheaper than ever before. And even though this was actually quite expensive in the beginning, the reasoning behind this sort of makes sense. And it's not just because they're vegan or because they don't want to consume meat. It's actually because of modern agriculture. Since 1960s, in the last 60 years or so, the global meat consumption has actually doubled. And as a result, this also has dramatically increased the amount of various greenhouse gases, and in the process has also created some major concerns both in terms of the safety of food and also the conditions on the planet. For example, approximately 15% of all of the global greenhouse gas emissions most likely comes from livestock. On top of this, modern techniques when it comes to, for example, raising cows, involve a lot of different chemicals such as growth hormones and a lot of antibiotics. And when it comes to microbiotics, it's believed that it's one of the main reasons we have so many different bacteria in a lot of different hospitals that are now extremely resistant to microbiotic treatment. Not to mention that generally, meat also contains quite a lot of foodborne pathogens and can be quite easily contaminated in a lot of other ways. None of which is a problem for cultivated meat. By removing animals from the picture, we actually suddenly don't have to deal with any of these problems. And though it might sound maybe a little bit naive, or extremely ideological, quite a lot of people out there think otherwise. There are currently over 60 different startups in existence, with quite a lot of them already in the process of developing incredible products. And since meat consumption is only going to be increasing in the next 2 decades by up to 50%, these startups are actually onto something after all. Okay, but I think most of us are probably not going to be spending $300,000 on a single burger. And so here, the cost is still a pretty big concern. Or at least it was, until so many startups started to create even more technologies and started to advance the field even further. For example, in 2020, an Indian startup known as Clear Meat claimed to have cultivated chicken meat that only cost about $10, while the slaughtered chicken would actually be a little bit more expensive. In this case, by using a donor chicken, they're able to produce 3 different types of cells from these chickens, including the muscle cells, the structure cells, and also the fat cells, which are essentially then grown in various bioreactors, kind of similar to how we produce beer. But instead of beer, we get chicken meat. Other startups, like MeatTech, or MeatTech I guess, use the idea of bioprinting to create something that has even more structure and thus more texture, with their overall product having a lot more quality, but also slightly more expensive price. But their goal is to have something like 6 euros per kilogram by 2030. But even compared to that burger from 2013, the cost in 2020 was already hundreds of times cheaper. Using the technology they have right now, it would actually cost them just a few $100s to produce something like this. And then we have companies like Eat Just that actually got an approval from countries like Singapore to sell cultured animal products like these eggs you see right here. With 1880 in Singapore becoming the first ever restaurant to actually physically serve cultured meat, with the chicken meal costing approximately $20. Still, I guess maybe a little bit more expensive than, let's say, KFC, but as I mentioned before, the prices are constantly going down. Other startups went even further. They basically went to space. The Israeli Aleph Farms became the first ever startup to produce cultivated meat on the International Space Station and even 3D print it in zero-G conditions, which theoretically means that astronauts might be able to produce their own meat in the future. So if you thought Star Trek replicators might never happen, this particular startup just took us a step closer. And interestingly, all of these advances mostly happened in the last 10 years or so, with all of this basically advancing exponentially. Which is really intriguing because back in 2008, there was $1 million contest for someone to create fake chicken using cultured meat by the year 2014. Nobody won this back then, but 10 years later, we get this. Not just chicken meatball, but a mammoth meatball. And so let's I guess talk a little bit more about this as well. Now in essence, the technology here is still very similar. It involved cultured cells that were grown on various scaffolds to create something that resembled meat with the right texture. But the difference in this case was the types of cells that they used for this particular idea. Because as I mentioned, there are so many different startups already, and because all of them are pretty much competing with one another, the company decided to introduce an extra step. They basically introduced a genetic engineering technique where they took some of the publicly available DNA structure that was discovered in various mammoths, and then inserted this DNA structure, along with the additional inserts from the elephant DNA, into the cells from sheep, and specifically into the muscle cells that could then be multiplied to produce all of the necessary meat. And so by using those muscle cells, this would give this meat very similar structure and very similar texture to typical meat we eat. And in order to give it that unique meat taste, they decided to insert the protein known as myoglobin that was discovered in the DNA of those mammoths whose frozen bodies were found in the last few decades. And if for some reason one day you want to actually try this yourself, or at least find that DNA from the mammoth, all of this is publicly available in the National Library of Medicine, with a specific link right here that's also in the description below. And so anyway, myoglobin from a mammoth, additional DNA from the African elephant, inserted into a sheep cell, eventually resulted in these very unique cells. Although not necessarily truly mammoth cells, these were really hybrid cells. And once all of this was cultured into enough material, it was then turned into an actual physical meatball. Apparently cooked to perfection and turned into this culinary masterpiece. Or basically what you see right here. Now this does look sort of interesting and maybe kind of delicious, but this is really not made for consumption, at least for now. First of all, obviously nobody really knows if it's safe yet. Second of all, because this was already in a museum for a while, it might have gone bad by now. And third of all, even though cultivated meat is already legal in some places, cultivated genetically modified meat is definitely not. Nevertheless, according to the scientists behind this, when the meatball was cooked, it did smell kind of like the crocodile meat. But how exactly it tastes, nobody knows. And we're probably not going to know for a while or ever. Because the point here was to really draw attention to the idea that this technology is possible, and that cultured meat is definitely becoming a reality, and possibly even a better reality. At some point potentially cheaper, definitely more sustainable, and environmentally friendly. Although I guess the more eccentric point they're trying to make here is that this meat will never be the same as regular meat. It's always going to be that extreme meat that some people might like and might really enjoy, but quite a lot of people probably will not. Nevertheless, because this is a design meat and can actually be created in a lot of different ways, both the taste and the nutritional value can be modified as needed. In other words, imagine having a meatball that's basically designed to be extremely healthy, but also actually taste the way you want it to taste. And in some sense, this does sound kind of exciting. But there's also obviously the question of ethics. First of all, is it even ethical to create something that's already extinct? Second of all, even though this is cultured meat produced from various cells, was it ever alive? Was it ever killed? This might not matter for most of us, but this might matter for some people that are, for example, religious. In certain religions, animals have to be killed in a very specific, respectful way before they can be consumed. Ironically, both the Jewish culture and the Muslim culture already have an answer to that. It really depends where the original cells came from. If the animal whose cells were used to produce this was killed in a way that's appropriate, in that case, Jewish or Muslim people can definitely consume this. But that's maybe going a little bit too far. Let's focus on baby steps. When exactly is this going to be affordable and possibly available near you? Well, at the moment, you can definitely get a lot of this in places like Singapore, and maybe even certain places in Europe, because apparently this meatball right now is in Netherlands. But it will definitely take some time before all of this becomes extremely affordable, even cheaper than regular meat, and it will probably take a few years before all of this is approved by the local authorities. And so the projected year for when this might actually become a reality in a lot of countries is maybe 2030. So at least 7 years from now. And by the way, if you're watching this in 2030, how are those meatballs? Is it as good as promised? Either way though, a pretty intriguing, somewhat clickbaity approach to promote the idea of cultured meat. And though I personally don't really mind it, as a matter of fact I'm sort of fascinated by stuff like this, not a lot of people are going to be happy with all of this, especially once it becomes a reality. But chances are we're not going to be eating mammoth meat. As a matter of fact, even that meatball is not really mammoth meatball either. This is a hybrid that includes a partial gene from the mammoth, but also includes things from the African elephant, and of course common sheep. And so I guess time will tell where all of this goes. Okay, today we're going to be discussing something a little bit more controversial and maybe somewhat unethical. And here we're going to be entering the, I guess, Victor Frankenstein territory. Brain-computer hybrid, where the scientists physically took human brain cells and then were able to train this unusual hybrid to recognize human voices, which apparently it was able to do. And so in this video, let's talk a little bit more about this particular study, what was achieved here and why it's important, and also briefly mention about some concerns in regards to ethics. But first, here is roughly what all of this looks like. A tiny brain tissue directly connected to computer architecture and directly able to communicate with it using input-output signals. But we've actually discussed something very similar in a video approximately 2 years ago that should be in the description, where the researchers used another brain cell hybrid to essentially teach it to play the game of Pong, which it was able to do very efficiently. But why exactly would anyone do this, and what is this for? And how is this different from all of the AI stuff we already have, such as the famous ChatGPT? Well, fundamentally these are completely different things, because our brain is ridiculously complicated in comparison to any computer we've been able to create. It contains nearly 90 billion neurons, each forming on average 10,000 connections. Recently, Australian researchers announced the creation of a supercomputer known as Deep South, trying to simulate human brain synapse operations by developing the most complex neuromorphic computer in existence. It's going to be able to perform 228 trillion synaptic operations per second, in some sense kind of similar to a human brain. But there's obviously a main problem with this approach, or I guess maybe a couple of problems. Supercomputer requires a lot of energy and can usually only function for a short period of time before encountering problems. A human brain only requires like a light bulb of energy, and as we're all aware, can function for decades. And so as much as we want to learn about the brain using computers, it's just always going to be extremely difficult, and we're never going to create something that's exactly like the brain. In terms of how fast the brain is in processing information, and in terms of how much information it can store inside of it at all times, there's just no computer on the planet that's ever going to be as successful as the human brain. And so because of this, some scientists decided to take a slightly different approach and start working with physical cells instead, with this process being much simpler and usually much more efficient. Here, the scientists rely on stem cells in order to produce a variety of different neuronal cells, with the stem cells in this case usually coming from a lot of different sources acquired ethically, with a lot of these cells then cultivated and grown into different cultures, usually with some sort of a purpose. And in the end they produce very small organoids that resemble something like this. Not really true brains, just tiny collections of brain cells designed to perform some kind of a function or designed to study something about the human brain. And in the last decade, since the invention of this process, there has been a lot of discussion if this is actually ethical, with the conclusion being that here there is really no personhood. This is not a living being, and not only does it not have enough complexity to acquire a personhood, it's also not connected to any other body parts and only has electrical impulses that it can then interact with. Although obviously some people are still going to be a little bit disturbed by this, but this has been used for a lot of different studies, and this is super useful for one thing we cannot do with computers: various neurological studies regarding neurological sicknesses. Anything from Alzheimer's to even things like epilepsy can be directly studied using these cells and can then be understood by studying them directly. That's completely impossible to do in a human being. Which is why this particular research is actually super important, and a lot of scientists are sort of excited about this. And because brains are so much more efficient than computers, some scientists decided to combine brains with computers to create something even more efficient in terms of learning. That's of course where we had that ping pong game from a few years back. And so combining the efficiency of the human brain with the ability to control things using computers, that's kind of the main purpose for a lot of this research. And so what exactly do we have here in this new study? Let's start with a few pictures. As you can see, it took approximately 3 months to grow this, with the brain cells organizing in 3 dimensions to complete an actual structure with a computer interface below it. And so in some sense this is a tiny tissue that's approximately 3 months old, that's extremely useful for studying how the brain works, how it develops, and what happens when things go wrong. But this device that they now call BrainAwave is a combination of the brain cells and the high-density microelectrodes. Here, electric signals send impulses into the neurons, where after processing the information, other neurons produce signals that can then be received by a separate computer. And the way it works is by signaling the neurons through the interface and by essentially sending the electrical signals inside this tiny organoid. The output is then read as the activity inside the organoid, something that's detected through physical signals from underneath the organoid. And that's because this computer interface also acts as a kind of a sensor. And even though so far this is pretty much exactly the same as what we've discussed previously with that ping pong brain, it's really what the scientists did afterwards that's kind of mind-blowing. By playing various audio clips, specifically 240 audio clips of spoken human speech, and specifically here it was a Japanese speaker making certain vowels, this unusual organelle, after just 2 days of hearing this, was able to identify a specific speaker with approximately 80% accuracy. And here he was able to make these predictions without any training whatsoever. Now this was obviously digitized and created as a kind of an electrical signal, so these were not actual voices, but it was still able to perform actually better than a lot of computers, because here the amount of inputs was very minimal, and it was able to achieve very accurate results in practically no time in terms of training. And because these organelles also have a lot of plasticity and are very adaptable, they can then be retrained for any task and can be used to study a lot of different responses to a lot of different types of training. But more importantly for medical studies, this is of course where you can do a lot of research on a lot of neurological disorders, but also conduct direct pharmacological studies using drugs in order to see what effects certain drugs have on actual physical human brain. Because here, by exposing this to some kind of a drug, we can then see how it affects learning in just a few days. Although obviously there are still quite a lot of limitations, with the biggest one being you have to keep this alive and healthy. It does require nutrients to survive, and will technically die if those nutrients are not provided. That's obviously not something we have to worry about too much when it comes to supercomputers. Another somewhat intriguing result is seen in the graph right here. This actually showed us that after growing for about 30 days, the organoid was not really responding much. After 45 days though, it was already producing certain signals, and after 60 days, it started producing a lot, in some sense representing the optimal level for these organoids. At 60 days, they seem to produce enough brain function to be useful for a lot of studies, but not enough to cause any ethical concerns yet. And so here it was actually interesting to see how fast these brains mature and how fast they acquire an ability to produce signals and to process information. But this whole scientific field, sometimes referred to as neuromorphic engineering, can obviously get very tricky very quick when it comes to pushing the ethics. With one company, BrainChip, even actually selling commercial kits that you can physically buy now and to some extent physically grow your own brain organelle. And that's of course where things get somewhat tricky. If this is commercially available for basically possibly anyone, who's to stop my neighbor next door from buying one and creating some kind of a super organoid that's possibly alive and functioning and conscious? Okay, I guess finances would stop him because these are still very expensive, but I think you know what I mean here. And so maybe commercializing this is not a good idea. This should still stay in the realm of research, and specifically medical research, or learning to understand human consciousness and of course human intelligence. But by commercializing this and possibly making this relatively easily available, I think over the counter at some point, that's what kind of makes me nervous, because we don't really know where this goes. And you probably remember a few concerns with potentially AI becoming conscious and becoming self-aware from a few years ago, which a lot of scientists argued against because we don't really think computer AI can become conscious yet. It's just not designed the same way. But a human brain, that's a different story. A human brain is designed to become self-conscious at some point. And so honestly, I'm not sure what to think about this just yet. Definitely important research, very important for a lot of studies on various neurological disorders so that we cannot study otherwise, and might lead us to understand human consciousness a little bit better, or it might also lead to my neighbor creating some kind of a monster next door. Yeah, not cool. Anyway, let me know in the comments what you think about this, but in terms of this research, very impressive, kind of mind-blowing, literally, and definitely something to maybe look forward to, hoping that nobody does something too crazy. Hello wonderful person, this is Anton, and when it comes to the origin of life on a planet, there are plenty of different mysteries and plenty of questions that cannot be explained even today. But today we're going to be discussing this one mystery that's maybe not as well known, and the mystery that's still completely unresolved. Something that we've known about for almost 200 years, and something that affects all life on planet Earth. But something that could potentially be somewhat cosmological in nature. But in order for me to explain exactly what we're talking about, and to basically then talk about the new study, here I have to start with the really important part of history, and a very well-known Frenchman that most of us already know, the microbiologist Louis Pasteur, who's technically known as the father of microbiology, and whose name is associated with a lot of different things. I mean, one of them is obviously pasteurized milk. But back in 1848, he discovered a very unusual phenomenon that he could not explain right away. Here he was studying what's known as tartaric acid, and this is actually something you can usually find in grapes and, in his case, in wine. I mean, he's French after all. Now, because he was also good at chemistry, he was able to synthesize some of the tartaric acid instead of extracting it from, for example, grapes. Now, by itself, this was already a pretty big achievement, but what he actually realized when he looked at it through various microscopes and by applying polarized light is that for some reason, both solutions, despite being tartaric acid, were producing entirely different optical observations. Now, the synthesized version was not producing any specific effects and was for the most part neutral. But the acid that was extracted from the grapes ended up shifting polarized light by just a little bit, suggesting that the actual solution from the grapes and the solution produced in the lab was not the same. The synthesized and the natural solutions were producing different optical observations. But in order to figure out what's actually happening here, he then decided to crystallize these liquids to see if they're going to produce the same crystals. And they obviously didn't. They produced something that was a little bit different and something that possessed different effects. Eventually he realized that he discovered something that we never knew about various organic molecules that today we refer to as chirality. Some molecules of tartaric acid were left-handed, but some were right-handed. Today more commonly referred to as dextro and levo in organic chemistry. But more importantly, what he actually discovered was the biggest mystery of biology. The synthetic liquid that he created contained both left-handed and right-handed liquids in equal amounts. Kinda makes sense. But the ones he extracted from grapes and other stuff were basically all right-handed. The synthetic acid had 2 different crystals, but the acid from grapes only had 1. And apparently at this point, this was his eureka moment. He exclaimed, "L'univers est dissymétrique." He discovered that life, for some reason, had a huge preference for chirality or for the handedness of molecules. Now, in case you've never heard of this before, chirality or handedness applies to most of the complex molecules that can be constructed in 3 dimensions. So basically here you can produce the same chemical formula, but the actual molecules are going to be different. They're mirror images of each other. And because of this, they actually do not have the same effects. Moreover, it turns out that all sugars on planet Earth are right-handed, whereas all amino acids on planet Earth are left-handed. And they cannot be exchanged for one another because they will not have the same effects. And this biological chirality, for about 180 years now, has sort of remained as one of the biggest mysteries. One of the strangest properties of life on planet Earth that really doesn't make sense. Especially because, except for one exception with bacteria, no life on the planet can use left-handed sugars. Surprisingly, there is only some types of ancient bacteria known to us that can physically change left-handed glucose into right-handed glucose in order to be absorbed later on, but they cannot digest left-handed glucose no matter what. Which also naturally raises a lot of questions. So can other-handed life exist elsewhere? Can we actually have RNA and DNA that's entirely right-handed, creating an entirely different type of life that's technically mirror image of us? And the answer to that is yes. We just don't know if it can exist on planet Earth, and it's probably not going to survive here for one simple reason. It's going to have nothing to eat. It cannot use any resources produced by life on Earth because we are basically left-handed. Or our DNA and RNA are left-handed, and our sugars are right-handed. Okay, I know it's a bit tricky and confusing, but that's basically how it is. And so even though both left-handed and right-handed sugars can exist in nature and are often created as both types, only right-handed sugars are used by life. And there's even evidence for chiral organic molecules in outer space in various nebula, although it's not clear which ones are more common. But for practically 200 years now, the scientists have been trying to solve this, and there are several sources for a potential solution. One of them is actually outside of planet Earth. By using organic samples collected from ancient asteroids, it could be possible to determine what sort of chirality is prominent here as well. One of the future studies of the samples from asteroid Bennu is going to be resolving one of these mysteries in the next few years. We don't really have the results yet, but they're coming soon. But the other way is by conducting various experiments here on planet Earth and trying to recreate this in a lab. So basically, can we actually create conditions where certain chirality takes precedence? And various experiments have been conducted in the past, with one conducted very recently. And so far, here are some of the potential explanations. The first one is, I guess, more or less simple. It could be an accident. Maybe sometime in the past, one of the molecules became more prominent, and eventually became used by most of the life that was developing. Other one kind of disappeared. And though it's a simple explanation, it does kind of make sense. The other explanations actually rely on something from outer space or on various physical elements that were present on early Earth. For example, in some experiments, scientists were able to show that polarized UV light coming from the sun can increase the destruction of one type of a molecule compared to the other one, resulting in, for example, more right-handed compared to left-handed molecules. Likewise, certain studies suggested that by being in a certain location in a certain nebula when the planets were just forming, it's possible that activity from very powerful stars resulted in huge amounts of radiation that would then destroy certain molecules, which would actually imply that there are probably several stars with several planets that all came from the same nebular cloud that all probably possess these right-handed or left-handed conditions. Likewise, a study from 2020 that we've actually discussed previously with a video in the description suggested that cosmic rays, and specifically muons, are often magnetically polarized in such a way that they're going to actually show a little bit more preference for the electrons from, for example, right-handed molecules compared to left-handed, which would increase overall interaction and also mutation, as well as destruction of potential DNA. So basically, in this case, the cosmic radiation itself could preferentially affect right-handed molecules as opposed to left-handed, although other studies have also proposed this to be neutrinos and not cosmic rays. So these are obviously more hypothetical, and this would be very difficult to prove, but they still kind of make sense. But now we have a new proposition that's actually a little bit more exciting than the ones previously, and this one involves an actual experiment that even shows the creation of only right-handed molecules. As always, the study in the description. Here it involves what's known as riboamino-oxazoline, also known as RAO. This is a very important precursor to RNA and is also either left-handed or right-handed. Specifically, it ends up producing cytosine or uracil. But like so many other sugars, it can also produce crystallized particles and can thus be studied a lot easier. And just like previously with Pasteur's experiments, it produces either right-handed or left-handed crystals. Although in this case, unlike previous crystals used, this one we know is needed for life. But only one type can be successfully used by life to produce RNA molecules. And so intriguingly, the scientists in this case were able to grow very specific crystals by placing them on magnetite. And when placed on magnetite surfaces, for some reason, the crystallization of this molecule was always 100% producing left-handed amino acid, the one needed for life. But they also were able to then produce opposite molecules also with 100% success by essentially changing the spin alignment or magnetization of the active surface. With the main implication here being that all of this could have been because of magnetic particles that were present all over the surface of early Earth. As a matter of fact, magnetite is often correlated with the existence of early life on the planet, with the main implication being that life might have a preference for handedness because of magnetite and magnetic minerals in primordial times. Although intriguingly, the scientists suggest that none of this seems to work in liquid muddy conditions, but seems to mostly work on a rocky surface containing solid magnetite. Nevertheless, here the discovery is really exciting. It produces an experiment that's easy to replicate that creates chiral organic molecules using very similar conditions to primordial Earth, which by itself is already quite groundbreaking. But it's obviously just the first step in trying to figure this out. As a matter of fact, we've actually discussed previous experiments that try to create synthetic life on the planet using various sources from bacteria, and here it would be interesting to find out if someone could potentially create synthetic life that's entirely mirror to a typical life we find here, and then it would be interesting to see what it's actually going to do. Now, the expectation here is that it's probably not going to survive. It needs to consume mirrored food, it also needs to have all its sugars in mirror format as well, but on the other hand, it's unlikely to be affected by viruses which would not be able to connect to it. And so what exactly this synthetic mirror life would do on Earth would be a very intriguing question to answer. Although intriguingly, some studies even propose that maybe if such life actually occurs on the planet, it could completely take over everything. Mostly because this bacteria, if it found some kind of a food source, for example algae that can actually use sunlight, would suddenly find itself without absolutely any predators or any enemies. Nothing could consume it, nothing would want to eat it, which could potentially lead to a major disturbance of the food chain. And though it does make a pretty cool science fiction story, apparently it has been explored many times, including Star Trek, and more recently in the Fantastic Four. So this is definitely a concept that has been explored previously and scientists have thought about before, but it's not a concept that we still understand very well. But more intriguingly, when it comes to astrobiology, the scientists would love to find out, assuming that life exists out there, is life actually mirror anywhere else, or is it always going to have same-handedness as life on planet Earth? And if so, why?
Hello wonderful person, this is Anton, and today I wanted to discuss a somewhat challenging topic to cover in a single video, so it's probably going to have several parts. And the topic is, I guess here on the screen, bacteria. But actually not just any bacteria. Bacteria and a lot of other microbes living inside of us, our gut microbiota or gut flora. Different microbes living inside of us and helping us to do quite a lot of different things. Because by studying these microbes, we can actually learn to understand ourselves better, and we can try to answer the question of, who exactly are we? Who are you? Who am I? Well, I guess I'm Anton, and you're the wonderful person, but that's not really what I'm asking. I'm asking, who exactly are we in terms of the actual cells in us? Because as we study these microbes, we realize that they play a huge part in our lives. They technically create us as well. And you might have heard the expression, you are what you eat. Well, today you're going to find out that not only is it actually correct, but in reality it goes way beyond that. The microbes inside of us are us, and we are sort of them. And so this whole concept is a little bit metaphysical, but kind of exciting as well. See, she's excited, and you're going to be excited after you learn all of this as well. Because one way of looking at our relationship with our bacteria inside our guts is to actually consider ourselves as a kind of a generation ship. Basically, a spaceship traveling through space for many years. And inside of the ship, there are inhabitants. There are different astronauts doing all sorts of work in order to maintain the ship itself. Now, replace the ship with yourself and replace the astronauts with the bacteria and a lot of other microbes living inside of us. And that's a kind of a relationship that we have going between us. You can't really have one without the other, and both are directly responsible for protecting and guiding one another. But let's start with the basics and with the baby steps. So for many decades now, the scientists were aware of so-called gut-brain communication, the direct communication between the brain and the small brain inside our guts. Both of them seems to have an influence on one another, with the gut actually sometimes having more influence on the main brain. And the gut itself also serves as the home to a lot of different microbes. Here we're talking about different bacteria, different types of ancient bacteria known as archaea, and even certain types of fungi, mushrooms. For example, this fungus right here, known as Candida albicans, is a pretty common fungus to be found in our guts. And though a lot of them do cause certain issues, a lot of them also probably play some kind of a role we still don't understand. And this is obviously not just true for humans. Many different species, even insects, have a lot of different bacteria living inside of them, helping them to some extent. But it's really in the last decade or so that we started to discover how incredible these bacteria and these microbes are in terms of providing a lot of functionality. For example, we know that they definitely protect us against different pathogens that would otherwise invade our system pretty easily. They also tend to protect us from various environmental toxins. Just like the colonists on that ship, they are also responsible for maintaining and repairing a lot of different things inside our guts. They're also responsible for providing a lot of support in terms of metabolism. They tend to digest a lot of things that would be indigestible otherwise. And because of this, about 60% of the waste that we produce, or basically our poop, is essentially dead microbes. Microbes that digested stuff that would not be digestible otherwise. But more recently, the scientists have also started to realize that they also play a huge role in various types of human immunity, and also seem to directly influence a lot of things in our brain, including, as you can see from this image, affecting sleep quality. And when it comes down to it, our guts essentially represent the highest microbial density of anything anywhere on the planet. There are approximately 300 to maybe even 1,000 different species living in us, with a lot of them still being discovered and a lot of them not really being understood at all. And what's even crazier, and that's actually why I wanted to start with this whole spaceship analogy, is that in terms of genetic representation, if you were to combine all of the genes from all of these bacteria, they would actually have approximately 100 times as many genes in total as there are in human genome. So genetically speaking, it's actually not wrong to assume that we are basically these generation ships for these microbial colonists. Their genes seem to be way more important and way more numerous than our own genes. And they're also obviously extremely ancient. Many of them seem to have existed on the planet for billions of years and only have adapted to live inside of us, possibly within the last few millions of years or so. But I guess what's less intuitive is that many of us, and actually all of us, have very likely different types of bacteria and microbes living inside of us. As a matter of fact, at least one study was able to discover that different cities seem to produce different types of microbes inside the people living in those cities. So there's actually a kind of a footprint or bacterial footprint depending on where you live. And the composition of bacteria inside of us has actually changed quite dramatically over time as well, especially as the diet changed and as we enter the industrial age. And this is a super important part because a lot of the modern sicknesses and diseases that humans tend to have are actually the result of either lack of or excess of certain bacteria in our guts. And all this is a result of the Industrial Revolution to some extent. For example, this study right here that was released last year was able to analyze some of the oldest microbes discovered in some of the ancient poop around the planet, analyzing the diversity of different microbes as far back as 2000 years, and specifically looking at the genomes of bacteria that used to live on the planet, in the process discovering that some of these bacteria that used to be in us were completely new to science. In other words, they were no longer around. They seem to have disappeared for one reason or another, whereas the modern microbiome contained a lot of different antibiotic-resistant microbes, but overall had much lower diversity of microbes as well. So basically we had fewer bacteria, and many of them were quite resistant to antibiotics. At the same time, discovering that there is a direct link between certain types of bacteria in our bodies, in our guts, and certain problems like, for example, diabetes or obesity. In other words, these little guys living inside of us seem to directly affect our health and seem to keep our bodies healthy, which seem to be very prevalent in ancient humans, the humans that used to eat a very diverse and very different diet compared to today. But interestingly, the bacteria of these ancient humans were somewhat similar to the bacteria of various native communities around the planet, although those unknown bacteria were still missing even from the modern humans living in various communities. And so the assumption here was that as we moved closer and closer to the so-called grocery store diet, we lost a lot of different nutrients that used to support those bacteria that very likely played an important role in protecting our bodies. For example, the degradation of various types of mucus in the colon is often associated with problems like Crohn's disease, celiac disease, or ulcerative colitis. All of this bacterial in nature, and all of this very likely connected to the modern diet. None of this was a problem back in the days. On top of this, some of these ancient microbes also had much higher numbers of so-called transposases, a type of an enzyme that usually is responsible for replicating elements of DNA or essentially helping various bacteria adapt to changing conditions. And so in other words, because of the ease of access of various nutrients in modern diet, a lot of the bacteria now has sort of become a little bit lazy and became less efficient in adapting to new environments. Something that's expected because of the way that modern diet works and because we generally tend to eat the same thing over and over pretty much every week. Now this is not the case for some of the ancient communities and some of the modern native communities where the diet changes pretty much every day. Or at least in more agricultural-based communities where basically you eat what you grow. Now, that's of course one of the main discoveries, but there were some other discoveries that were much more surprising, specifically based on various studies involving mice. And in this case, by studying the gut bacteria inside mice, the scientists actually discovered something really interesting. By doing what's known as the feces transplantation, or essentially taking feces from one mouse and putting them into the other mouse, something that's done in medicine already, they were able to successfully reverse aging and problems related to aging in different older mice. And more specifically, they were able to reverse a lot of the cognitive decline and a lot of cognitive problems associated with being old. Once again confirming that the healthy microbiome seems to directly support not just the digestive health or the immune system, but also support the function of the brain as well. And that's of course a super important discovery because even though it's extremely difficult for us to change our DNA or to somehow reverse aging by, for example, changing things inside our body, In this particular case, this study suggests that by transplanting microbiome from the younger individual into the older individual, it might become possible to actually improve the health of a certain individual. Going as far as discovering that even their brains seem to have repaired just a little bit, which did improve cognitive function in these older mice. With at least one bacteria, this one here known as Enterococcus, very likely being responsible for a lot of this because it's known to be quite abundant in the younger mice and seems to be absent in some of the older mice. But just the fact that it actually seems to have changed the brains of these mice is already a huge discovery. And because in humans something similar is already used to treat conditions such as different allergies or, for example, irritable bowel syndrome, it's probably not long before the doctors potentially start using the poop transplant from younger donors to reintroduce healthy bacteria into someone's gut and to then help them improve health that way. And all of this goes along with some of the other studies that have recently also discovered that whatever is happening inside our guts is directly affecting our brains and potentially even changing our personalities. And so this so-called gut-brain axis is extremely potent in affecting everything about us. A lot of the chemical signals that are generated inside our guts affect our brains, and many of these signals will generally be produced through the activity of various bacteria. And going back to mice again, one of the experiments used various types of transplants from people with what's known as the major depressive disorder, MDD. And when these samples were transplanted into mice have also developed somewhat similar and unusual symptoms. A lot more anxiety, a lot more depression. And so going back to that girl with the pizza, you are what you eat after all. But it seems to go much further than that. It seems to be way more potent than we ever thought. You're not just what you eat, the bacteria inside of you actually define who you become and how you feel. Not to mention that it's also connected to the quality of the sleep you get, it's also directly connected to various anxiety disorders, and it also seems to be connected to a lot of depression and other mood disorders that we're still trying to learn about. And since the microbial composition inside of us tends to change depending on the time of the day, it also means that our guts are exposed to different types of metabolites produced by different microbes during different times of the day, making all of this connected to your sleep quality and, of course, the idea behind the circadian rhythm. And there also seems to be a connection between different types of personality traits and specific microbes living inside of us as well. Certain microbes tend to produce certain personalities, something that was discussed in this study that was released only a few months ago. With the principle being relatively simple, certain bacteria will leave behind certain metabolites or certain types of chemicals that can then generally influence the so-called brain-gut axes, producing certain signals that would not be produced otherwise, with each type of the bacterium being associated with a very specific personality trait. Which implies that depending on what you eat, you're going to be encouraging the growth of certain types of bacteria, which in essence changes your personality with time. None of this is going to be instant, and it's going to depend on the balance inside your microbiome, but all of this seems to be correlated. Which takes me back to that original point. You are what you eat to a much bigger extent than we ever thought. It seems to affect your personality, it seems to affect your cognitive state, and it probably has a lot of other effects as well. The gut microbes seem to directly affect who we become with time. But that's of course just preliminary findings from just some of the studies in the last few years. There are still so many unanswered questions. For example, what effects would the microbiotic have if it suddenly ends up eliminating most of the bacteria responsible for certain personality traits? On the other hand, what about dramatically changing your diet? How quickly will the personality and everything else change afterwards? And so obviously all of this is still in its infancy, and there are so many new things to learn about ourselves and about our little passengers, the microbes. But then again, there is still that question that I cannot answer. Who are we? Who are you? Who am I? Are we basically just these generation ships controlled by the bacteria who seem to be extremely efficient at living inside of us and represent some of the richest microbiomes in the world? Or did the bacteria basically occupy us without asking and have become permanent residents even though we never really wanted them to be there?
A long time ago, approximately 4.3 billion years ago, singular cellular life established itself on the planet, and it thrived on Earth for billions of years. But at some point, something else happened in the history of the planet when some of this life decided to combine into something larger. Single cells, started to form larger and larger structures, eventually developing into multicellular life. With some of these structures becoming permanent, some of these structures acquiring different functions, and eventually evolving into some of the first primitive animal life on the planet. Life that contained billions and even trillions of cells, differentiating and creating specific organs, and eventually evolving more and more complexity. Or in essence, sometimes in the past, we had this very unusual transition from basically successful single cells into more complex multicells. And this was of course the creation of the first multicellular life. Or so the theory goes. We obviously have no actual proof or evidence for any of this, mostly because none of this would survive billions of years of being stuck inside the fossil. But because our planet still has single cells, and because we have multicellular life like ourselves coexisting with single cells, it's the only explanation that currently makes sense. But how exactly did this change from micro to macro happen? How did one cell become many, and what led to the formation of this unusual structure that, I guess to some extent, would not really make sense at first? Single cells are usually very successful by themselves. Now, it's a difficult question to answer, and especially a more difficult question to answer when it comes to the timeline, but it's a question we can try to answer by conducting experiments in present time. And it looks like this recent experiment might have provided some extremely essential answers for how this might have happened. Hello wonderful person, this is Anton, and today we're going to be discussing one of the potential answers for this unusual phenomenon of single cells becoming multicells, or a crucial step in the evolution of complex life on the planet that up until recently was somewhat difficult to answer. A recent experiment was actually physically able to recreate a formation of multicellular life. Not a really complex one, but actually somewhat large in size and even visible to a naked eye. And somewhat related to the Fermi paradox videos on the channel, to some extent this also relates or provides an answer to the idea known as the Great Filter or the Great Filter hypothesis. We know that for some reason life on the planet went through this extreme change where a lot of single cells on planet Earth preferentially started to evolve into multicells, eventually leading to organisms like us. And it's actually super important to figure out why this happened in order to figure out if it could happen on another planet. An alien planet out there would have to have very similar conditions to planet Earth, or at least similar limitations, in order for multicellular life to occur. And so what exactly did the scientists recently discover? Well, as always, you can find the links for all of this in the description below, but in a nutshell, this particular study focused on studying something that's already been studied for quite a long time in a lot of different similar conditions. Here we're talking about this. This is common yeast. The stuff that we use in baking, and the single-cell microorganisms that are technically eukaryotic, which makes them more complex than bacteria, but in essence are also members of the fungus kingdom, basically mushroom kingdom. And unlike bacteria, they were only developed a few 100 million years ago, so these are recent arrivals to planet Earth compared to more ancient life. Now, one thing that makes yeast somewhat interesting is that it does have a tendency to collaborate or cooperate with bacteria as well. Here's an example from the drink known as kombucha. And in some cases it forms relatively complex colonies as well. And it's obviously also relatively easy to grow, and humanity has been growing yeast for thousands of years, since some of the early bread and of course some of the earlier beer. And so because it's so easy to grow, for the past few years some scientists in certain labs wanted to see if they can actually evolve yeast a little bit faster. And so in some of the earlier experiments, they grew yeast in tubes and watched some of the cells combine into larger chunks and sink to the bottom. And so as far back as 2012, they actually were able to selectively breed certain yeast cells to create larger and larger structures much faster. Or, just to rephrase this, they selectively bred yeast to form small multicellular structures with a lot more efficiency and much faster than regular yeast by selectively breeding it over several different generations of collecting yeast chunks from the bottom of these tubes. This was basic Darwinian selection at work, and this didn't really take long to produce larger chunks. As a matter of fact, according to the scientists behind the study, this only took approximately 60 days. And so after 60 days of breeding, most of the yeast would prefer to form larger structures compared to single structures. But this of course had no actual reason for happening, and normally occurred randomly, because instead of drifting apart, some cells would stick together, creating larger and larger structures. Either way, after just 2 months, they went from predominantly single cell to multicellular yeast that looked a little bit muddier and a little bit chunkier inside a typical tube. And obviously this was kind of interesting and somewhat intriguing at first. It didn't necessarily show us how multicellular life evolved afterwards, especially because at this point they basically got stuck. This was as big as these yeast cells would get. No matter what the scientists did, they would not get bigger than a few micrometers. And more importantly, they didn't actually have a very strong connection and would be very easy to break. And so not only were they kind of fragile, they sort of refused to grow past a few hundred cells. And so in that sense, this was not really a proof of anything. It didn't show us how multicellular life could have evolved, and just showed us that some cells can basically just stick together. But that's of course until the more recent study from 2023. The scientists did something else, and it really transformed everything. With this image alone, enough to show us the transformation. It went from just a few hundred cells to really large chunks visible with naked eye. Some chunks being 20,000 times bigger than anything from 10 years ago, and containing up to 500,000 cells, in many cases with specific function. And more importantly, three-dimensional structure that seemed to be more or less permanent and would not break very easily. And so what exactly happened here, and what did the scientists change in order to demonstrate all of this? Well, intriguingly, they didn't actually add anything. They took something away. By taking these smaller chunks and depriving them of oxygen, they suddenly created conditions where it was beneficial for yeast to not be alone and to be in larger structures. And keeping yeast in oxygen-deprived conditions for several months, they realized that all of these structures would grow larger and larger because it seemed to be beneficial for their survival. Within 600 days, they would be visible to naked eye. Here this is a few millimeters across. More importantly, a lot of cells in this case became much longer, strengthened the connection between themselves, and started to develop individual structures that would provide additional resilience, making these structures stronger than the wood, to some extent even resembling simple bodies. But more importantly, the cells within the structure now started to acquire different roles. Some cells grew really fast, some cells provided very rigid, very tough structure, yet other cells for some reason would self-destruct. In other words, this was a definitive sign of multicellular formation, same cells performing different roles in a much larger organism. Although this type of emergent behavior is actually quite common in nature. You can actually find another video on the famous slime mold in the description below, where this unusual single-cell organism starts to acquire a lot of complex emergent properties as it becomes larger and larger in size, to some extent even mimicking simple intelligence. And so seeing these emergent properties coming from common yeast is not unusual either. As a matter of fact, we believe that this is sort of a common feature for a lot of complex structures. As something becomes larger and more complex, a lot of unexpected emergent properties usually start to arise as well. What sort of properties is anyone's guess. Although I guess the more interesting question here is why exactly did some cells start to self-destruct? Well, here the scientists believe that maybe this is one of the ways they can create reproductive clusters. By releasing small fragments into the environment around themselves, they basically provide more nutrients, possibly allowing other structures to grow larger and larger as well. Although at the moment it's kind of difficult to determine. And intriguingly, after just 2 years, these structures also evolved an emergent property that allowed them to bring nutrients to the cell inside the structure, while at the same time getting rid of various types of waste. Basically, it managed to evolve a really rudimentary circulatory system. With all this basically happening as a result of that external pressure, lack of oxygen, and selective breeding, recreated entirely in a scientific lab. And though usually scientists assume that oxygen should be good for multicellular life, it's really the absence of oxygen that seems to cause life to come into larger structures and to form larger bodies. In an oxygen-rich environment, it's probably a lot more beneficial to stay as a single cell. And though obviously it doesn't show us how something extremely complex can form, it definitely demonstrates the formation of much more simple multicellular life, which in time could evolve complexity as well. And because we know that multicellular life on the planet evolved at least 25 times in the past, these new experiments and these new observations show us that maybe this is something that's very common across the entire universe, assuming that single cells exist there as well. And when it comes to Fermi paradox and the Great Filter, presenting some evidence that maybe this is not one of the filters after all. Maybe this is just something really common and could happen anywhere. Because it definitely happened in this lab in just over 2 years. And that's absolutely incredible. It's actually a super intriguing discovery when it comes to the evolution of life on the planet, and I'm sort of looking forward to seeing more from this particular lab, which by the way you can learn more about in one of the links in the description below. Hello wonderful person, this is Anton, and today we're going to be discussing some of the new discoveries in regards to hibernation, or in regards to suspended animation, or basically the state of minimal activity and metabolic depression that's present in a lot of different species, especially during relatively difficult months of the year, with I guess the most common example being a bear. But in this video we're going to be talking about some of the new advances and new experiments that might have discovered that this is also potentially possible in primates, achieved through a very specific chemical trigger by basically activating certain receptors in the brain. But also generally talk about some other facts and other discoveries in the last few months. And by the way, this is technically part 2. The part 1 of this video is available in the description below. Although in this case, this younger Anton was specifically discussing the idea discovered in some of the experiments conducted in Canada that focused on various types of squirrels that actually have these unusual bacteria inside their guts that could also serve as the potential trigger or actually potential support for hibernation in humans as well. But the study we're discussing today is a little bit more intriguing. First though, let's start with a few misconceptions. And naturally with a topic like this, there are going to be quite a few to cover, but I'm only going to mention some. For example, when it comes to bear hibernation, it's kind of surprising that it's only recently that the scientists officially confirmed that bears do hibernate. In the last few years, it wasn't really clear if bears truly hibernated or if they were just sleeping in a very different way. But turns out that they are able to, for example, recycle their own urine during their prolonged sleep, and are also able to lower metabolism and body temperature for a prolonged period of time. So in other words, they do hibernate after all. But the reason this was even questioned is because other animals, like for example bats, hibernate with a lot more extreme effects. They can generally lower their temperatures even lower, and they actually have quite a lot of recycling going on the inside. And even though so many different mammals are able to hibernate, for some reason, the same is not true of primates, monkeys, apes, humans. And as a matter of fact, up until extremely recently, the scientists thought that primates for some reason do not hibernate at all. But as always, there was one exception discovered not so long ago. And this exception is a lemur on Madagascar. It's known as the fat-tailed dwarf lemur, and it's able to hibernate for up to 9 months. But more intriguingly, it hibernates for completely different reasons. Most animals known to us hibernate during winter, and usually because the conditions are really cold and there is just not enough food around. But these little guys do it for almost the opposite reasons. They hibernate when it gets too hot, and they seem to do so to avoid drought, suggesting that hibernation is not exclusively an adaptation to low temperatures, making these guys a kind of a double exception. They hibernate when it's hot, and they're also the only primates known to us to hibernate every single year. Intriguingly, that study that analyzed their hibernation patterns even discovered that depending on the outside temperature, and more specifically, depending on the temperature inside of their habitat, they will also experience different sleep patterns. Whenever the temperatures are much higher, they tend to have what's known as the REM sleep. They basically start dreaming. We can only guess what they dream about, but probably food and water. But when it gets a little bit colder, the sleeping patterns switch completely. Their body enters lower metabolism, and the REM sleep stops completely, along with a lot of other body activities as well. Which of course makes this animal an extremely important case study for a lot of hibernation research. But the more important question in hibernation has always been, okay, so a lot of animals do it, but can humans do it too? Or have we done it before, thousands of years ago, when we were a lot more primitive? Well, we can't really answer that second question yet, but we can try to answer the first question, and we can do so by possibly learning if we can hibernate other primates as well. Now here I actually wanted to mention that, as of today, there's no official case of humans hibernating that's been proven scientifically. There are, however, some anecdotal cases, like Carolina Olsen, also known as the Sleeper of Okno, who allegedly hibernated for 32 years. But there are a lot of things about her story that don't really add up. We'll maybe discuss her story sometime later. Either way though, it's not particularly interesting scientifically speaking. There's also some earlier research from the 90s that claimed to have induced hibernation in animals by injecting them with blood from other hibernating animals. But it was not very easily reproducible, and so the actual conclusions were not super clear. But the main question is, can we actually induce it? Can we trigger hibernation in a more complex animal like a primate or a human? Well, some of the earlier research from the 90s claimed to have induced hibernation by injecting the blood from hibernating animals into other animals of the same species. But the results were not reproduced by everyone, so it's not clear if this is actually something that works. And more importantly, it's not clear what's actually causing the hibernation itself. Although quite importantly, some of this research discovered that there are different substances in the blood of hibernators that tend to produce something that protects organs for a long period of time. And some of these substances were even used to prolong a life of isolated pig's heart. Which means that, in theory, we could use these substances for transporting different organs during transplantation. The scientists have suggested this could increase transplantation time up to about 20 hours. And moreover, similar substances seem to kind of exist in primates and of course humans, which implies that our bodies technically can hibernate, with the question of course, how? Is there actually a way for us to maybe trigger it? And that's exactly what this new study was about. It was able to chemically trigger a state of hypothermia by activating specific neurons in the preoptic area, also known as POA, of a group of different macaques. Animals whose brains are, to some extent, somewhat similar to ours structurally. And though obviously this wasn't a true state of hibernation, this was to some extent its cousin, or basically the beginning step of hibernation. And this was done by using the crab-eating macaques, whose brains were modified to have a very specific receptor inside hypothalamus, the tiny organ sort of in the middle of our heads, responsible for various metabolic processes, but also responsible for things like body temperature and hunger. And then these macaques received a very specifically designed drug that would act on those receptors in hypothalamus, activating preoptic area cells. In other words, these drugs were specifically meant to trigger only those cells. And then by performing different types of analysis, including MRI, they were able to confirm that the activity inside these cells induced hypothermia and a lot of other effects, resembling initial stages of hibernation. Overall, their core temperature dropped dramatically, their heart rate also slowed down, they started to exhibit effects very similar to what we see in someone going through hypothermia, and they even started to shiver. Okay, so maybe this doesn't sound very important yet, because basically it's like making these monkeys really cold. But there were definitely a lot of changes in the blood and changes in various substances that sort of resembled initial stages of hibernation in other animals. And so does this mean that we finally discovered what triggers it? Well, not really. As a matter of fact, the scientists mentioned that this is actually a little bit complicated. They performed a similar experiment on mice, and in mice, the heartbeat also slowed down, their metabolism dropped dramatically, and they kind of started to conserve more heat. In other words, in mice, it might really lead to hibernation. Yet in these macaques, the effects were almost the opposite. Their activity increased dramatically, and so did their heart rate. As I mentioned, they started shivering, and they basically started to attempt to create more heat. I guess a natural reaction to being cold. With the implications being that thermoregulation and hibernation in primates is definitely a much more complex topic than it is in other mammals such as mice. And though the scientists might have identified the first trigger to potentially induce hibernation, it's quite likely that it's not going to be as simple as just triggering a few cells. It's a lot more likely that this is going to be a super complex process, and we're obviously not even close to understanding what needs to be done for a human to hibernate through, for example, a very long space mission. Although in this case, the actual answers are probably still going to be in hypothalamus, as this is technically the main organ responsible for a lot of activity when it comes to regulation of body temperature and metabolism. And as I mentioned in one of the previous videos, this is going to be extremely important for any prolonged space travel. It would dramatically reduce the amount of material that we need to bring. It would also reduce the psychological and even physical pressure from being constantly awake and working in space. And more importantly, would also preserve our organs. For example, the muscles will not atrophy as much if the human is actually hibernating. And the same is true for our heart and a lot of other organs. Which means that hibernation research for space travel is probably one of the more important things we can focus on if we actually realistically want to send humans to Mars. Otherwise, after 6 to 9 months in space, a lot of astronauts unfortunately might develop too many health problems to be of any use for any mission on Mars.
So definitely pretty exciting research and pretty exciting discoveries. But naturally just the first discoveries we've made so far, and probably the first we're going to be making for the next few years. This type of research is extremely difficult to conduct, and especially because it would involve humans, and so it might take a very long time before we have a working solution to induce hibernation in human beings. But maybe this right here is the first step.
It's that time of the year! Halloween special! And stuff. But this time with even more science than ever before. And it just so happens that I have a perfect topic for Halloween. The penultimate topic, if you will. It's about Draculla, as you can probably tell from the title of the video. More officially known as Vlad the Impaler, or Vlad Tepes in Romanian. And that's because of his reputation for basically impaling people on wooden stakes, especially if they crossed him or if they disobeyed him directly. But more specifically, we're going to be discussing some of the extremely recent discoveries about the actual person of Vlad the Impaler, because in a recent paper, scientists were able to use some of the papers that he used to write on to basically analyze his DNA, discovering that he actually had an unusual disorder that potentially made him cry blood, adding to the already mysterious persona that he had back in the days. And so let's discuss Vlad Draculla a little bit more from a historical perspective, find out how he got the reputation he has today, and talk about these recent discoveries. But first, I guess, the obvious. A lot of the reputation he acquired back in the days was really because of the time and the place where he was one of the rulers. His father, Vlad Draculla, was the ruler of a small kingdom of Wallachia. Today it's part of Romania, but back then this was a kingdom that was essentially bordering the growing and developing Ottoman Empire. Around this time, the empire was focusing mostly on expanding northwest and was basically conquering one European kingdom at a time. But Wallachia resisted, and so in order to basically make them a vassal and to avoid further confrontation, Prince Vlad, along with his brother, for many years were held as hostages in the Ottoman Empire in order to secure their father's loyalty. And the entire region during this time experienced constant warfare from various factions trying to control the land. At some point, Vlad was actually able to become the ruler, which did involve crossing a few people here and there, but what made his strategy a little bit different is that he decided to use fear as the weapon. During a conflict with nearby Transylvanian Saxons, he plundered some of the villages, taking a lot of prisoners back to Wallachia, where he essentially had all of them impaled afterwards in order to kind of demonstrate what happens if someone crosses him. And it was so effective that he basically started doing this everywhere. But at this point, the Ottoman Empire was growing larger and larger. Here's actually what it looked like within just 80 years, and because of this expansion, they were essentially trying to make sure Wallachia either becomes part of the empire or at least pays annual tribute. And so one of the Ottoman sultans, Mehmed II, ordered Vlad to start paying tribute around the year 1462. But instead, he captured the Ottoman envoys, had them impaled, and then attacked Ottoman territory nearby, further impaling thousands of people. And this really solidified his reputation as Vlad the Impaler. Apparently he might have impaled up to 80,000 people during his reign, but the reign itself ended pretty quickly as soon as Ottoman Empire decided to strike back. And so in 1463 he was captured and held in captivity for approximately 12 years. And two things happened during this time. First, some of the first stories about his cruelty started to come out in a lot of different European countries, mostly because he was seen as a kind of a protector of Christian faith and was pretty successful fighting the Ottoman Empire. And second, near the end of his captivity, he also started to write himself, with some of the letters still preserved today. This one was written in 1475. And even though he was released in 1475, he didn't survive long and perished 2 years later. But at this point, various stories about how cruel he was and how he basically reigned with fear started to spread everywhere, making their way to Western countries where something else happened around the same time. In 1400s, in Germany, Johannes Gutenberg invented his famous press. And it just so happens that one of the first bestsellers in Europe, printed in these new presses, was basically about Vlad the Impaler. At this point, it was bigger than the Bible, and probably one of the most read stories in most of the Western European countries. And today it's actually believed that it became so popular because of the cover. This was literally on the cover of the book, and was extremely different from anything else at this time. It presented a ruler who was dining in a field surrounded by impaled bodies, representing a horrific scene never before seen in any books previously. This particular image is from the edition published in 1499 in Nuremberg. Here's how he was portrayed in some of these German books, with most of the book dedicated to his cruelty. But naturally, he lost the war. The Ottoman Empire reached its peak approximately 80 years later, and then shifted its attention to North Africa. And so despite these early publications and, I guess, early success, this viral sensation eventually got replaced by other stories, and he was to some extent forgotten for some time. And that's of course until Bram Stoker started to do research for his novel Draculla. And he actually wanted to find some kind of a historical connection to various Transylvanian superstitions involving vampirism. He found out about these superstitions by reading articles by Emily Gerard, who essentially described various Transylvanian practices, including the fear of unusual creatures that were believed to suck blood. These stories might have inspired a lot of other artists, including Edvard Munch, who literally has this painting called Vampire, and this was painted before Dracula came out. There are actually quite a few vampire paintings around this time, so this clearly was a very popular topic in the late 1800s. And during his research, Bram Stoker somehow discovered the stories about Vlad the Impaler, eventually turning this into one of the most famous horror books of all times. Although today historians believe he might have not actually done a lot of very thorough research, because first of all, in the book, there doesn't seem to be a lot of reference to the actual Vlad the Impaler, with a lot of other references, kind of confusing various regions, various belief systems, and more importantly, making them the leader of Transylvania, even though he was technically the leader of the region they were at war with. Transylvanians were some of the first victims of his impaling, and Wallachia was an entirely different region. Nevertheless, this was technically the birth of Count Dracula and the birth of vampires as a phenomenon. But it really wasn't until 2023, or basically just a few months ago from when I'm making this video, that scientists finally discovered something else about Vlad the Impaler that nobody actually ever mentioned or knew before. And it's not entirely clear what this means just yet. And in this case, this was done using a very intriguing technique, involving handwritten letters by Vlad Tepes himself. And here the point is that when we write by hand, it's quite common for the person doing the writing to place their palm on the paper, making the palm rest and rub on the paper as you write. And it turns out that this actually transfers quite a lot of organic molecules from your palm down into the paper, which can then be preserved inside the paper for a very long time. And so knowing this is kind of what the researchers behind the paper in the description wanted to explore. By applying and removing ethylene vinyl acetate, and by then using mass spectrometry, they discovered residues of over 500 different peptides, with 100 being of human origin. Peptides are basically very short chains of amino acids, very often containing parts of proteins, which can then be used in genetic analysis. And by then conducting the analysis of these proteins and peptides from 3 separate letters written by Dracula that all contain these proteins, they were able to discover something really unusual. First, they discovered signs of what's known as ciliopathy, a genetic disorder that often creates problems for various cells and for various organs. There's no one disorder that it can cause, but there are a lot of different syndromes and potential disorders that can be caused by ciliopathy long term. It's not clear if this manifested in him, but it very likely did. They also found evidence of some kind of an inflammatory disease, potentially resulting in major problems with his skin and respiratory tract. In other words, he potentially had a major skin problem, along with maybe some kind of asthma. But more intriguingly was the discovery of compounds very often linked to what's known as hemolacria, an unusual condition that tends to mix blood with various fluids inside tear ducts, literally resulting in tears of blood. And that by itself makes this whole story oh so Halloweeny. I mean, if this is true, then this is a completely new level of Dracula we never knew about. Now, obviously none of this is mentioned in any of the chronicles about him, and I'm sure anyone who possibly even wanted to mention it ended up on a stake, but if this is actually confirmed, it definitely makes Dracula an even more intriguing person, more mysterious, and more scientifically intriguing compared to anything we knew about him before. And so, if these studies are correct, for all we know, Count Dracula, or Vlad III, Vlad Tepes, Vlad the Impaler, might have been asthmatic, had some kind of a skin problem, potentially cried tears of blood, and might have had some other disorders manifested through ciliopathy, which back then would definitely make him some kind of a unusual character. Some of these somewhat rare, mysterious disorders would definitely make him a somewhat unnerving person to be around. Unless I guess he never cried, which maybe he didn't. But anyway, definitely some really intriguing discoveries just based on these 3 letters, and something you can learn more about by reading the study in the description below. And hopefully this research leads somewhere else, because we might discover something else for next Halloween, so I get to talk about that as well. But until then, thank you for watching, subscribe, check out some of the previous videos on similar topics in the description below, support this channel on Patreon by joining channel membership or by buying the Wonderful Person t-shirt you can find in the description. Happy Halloween! Stay wonderful, I'll see you tomorrow, and as always, bye bye.